biochemical optical biosensors, plant nanotechnology, carbon nanotubes for bioimaging and all. You can see a whole list of publications related to all these very specialized area. There's a lot of scientific publication, around 49 peer-reviewed research articles, 14 book chapters, and he has edited 3 books. There are 8 patents to his credit, and he has delivered many invited talks in many international journals. So he's a very well-known international fame. He has guided 22 PhD students who has completed PhD under him. In 2012, the Society of Polymer Science Japan honored him with the Asia Excellence Award for his research in biopolymer. He is currently the editorial member of the Scientific Reports, one of the very famous journal under the Nature Publishing Group. Professor Shakti Kumar will be presenting here today on the Theranostics Nanomaterials Against Cancer. On behalf of the organizing committee, I welcome Professor Dr. Shakti Kumar for his presentation. Over to Professor Shakti Kumar. Welcome, sir.
Thank you, Anil, for a great introduction. And thank you very much to Sudha and Mohan sir for inviting me to deliver this talk. After this COVID-19, almost our travel and everything has been stopped. Otherwise, I had to be in US today for organizing MRS1 session. So anyway, nowadays we are doing everything through online. So the reachability is much more higher compared to talking to a very small group. So I would like to talk today regarding theragnostics or theranostics, nanomaterials against cancer. So this theragnostics or theranostics, what we call, because it's as therapy plus diagnosis together. So I would like to go into details in coming slides. So you can see here, with the slides which I have shared just earlier is a view of very good Fiji mountain. Recently it took the photograph of that one. Is really beautiful, and you can see that this is a countryside. Picture is very beautiful, and these are the situation now in Tokyo and nearby areas. So you can enjoy all these things nowadays. A lot of color changes and very beautiful. Nature is actually blessed with a lot of beautiful colors. But you have to enjoy all these things. You should have a very good health. That's very important. Even if you have a very some cut in your hand or leg or something, or maybe you have some pain, even if something is coming in very good in your eyes, in front of your eyes, you will not be able to enjoy those things. So it is very important. We should have a very good health. We call it as in Hindi or Sanskrit, it's a swastha. We should be that kind of mindset. Only then you will be able to enjoy all of these things. So in our ancient scriptures also, they have mentioned that getting a good health and good body is very important to enjoy your life. So that is very important. So, but we are going through a lot of problems and diseases, and many things comes to our body, and which creates a lot of problems to our mindset and our health itself. So one of the main problems is actually cancer. The problem is that this cancer is a ongoing, and prognosis, is a maybe some of the cancer, it can take up to 5 years, 10 years for till your end. That kind of things are happening. So the problem is that when you diagnosed with cancer, so the thing which we are coming into our mind, okay, we are going to see the imminent death in our front. So we'll not be able to enjoy. That is the thing. So even though many others are saying do yoga, that one, this one, to control your mind, many things are there, but still these kind of things will be lingering in our mind. So we'll not be able to enjoy our life. And always we will be under tension, even if we will not be able to give 100% whatever the things which we are doing. That is one of the main problems when we have these things are there. So what is happening nowadays when this thing is setting, and we'll come to understand that we have cancer. the total devastation, helplessness, fear, anxiety, all these things will come into our mind. What we have to do, we don't know what to do. So, but there are drugs which we have found many of the times. That kind of drugs actually will come like this. So treatment, its current method is actually carpet bombing. Put some toxic materials. It's actually, even though called as a drug, actually we are using some toxic material to kill the cancer cell. But while doing that one, normally what we are doing, we are actually devastating not only the normal cells, we are killing the non cancer cells, we are killing the normal cells also. So this actually it can prolong a little bit our life, but it doesn't mean that we can come out of this problem. So we will end up in a total devastation. This is the one main problem which is happening nowadays in the field of cancer therapy. So what is actually we are looking for? That is important. So we are looking for like a fighter, just like a seals, which can fight in the land, air, as well as in sea. So like perfect fighter at any places, and can take his own decisions at times, so that to kill and the target. And for that one, highly trained multitasking materials we should have to kill in the case of cancer, just like a seals, highly multitasking. So we need the imaging and to track and deliver the drugs only to the site, kill the cancer cells selectively, minimize the collateral damage. That is, we cannot say that we can take away all, then we can nullify the damage, but we can minimize it. That's one thing we can do. And provide immunity, as well as eradicate the possible relapse later. So these are the things which we are looking for in the field of nanodrug delivery. So in that way, we can say that therapy as well as diagnosis combined together. So we need theranostics materials. So in these kind of things, we have to take all these materials. This is the why, it is becoming multidisciplinary work. We have to engineer some materials. We need to get highly biocompatible materials. We have to we need the targeting materials. We need drugs, how to put this drug into the materials inside and how to deliver it, and all these things we have to do. So that is why this become a very prominent research field, and we have to use many materials, many different from different fields. We have to utilize all these things to make a very good theranostic material, one piece to deliver the drugs. So it depends upon many of the properties, just like porosity will be there, roughness we have to sometimes we have to find out, and the shapes which we have to utilize some of the times, and many of the times we have to use nanoshells, liposomes, polymers, iron oxides, and all these things which we have to utilize for our different making different nano compositions for developing a theranostics materials. This is very important. So persons with different practical the experience have to come together and to make all these things. Then we can come forward with a very good materials. And because of this, we are also, our group is also working in different fields in the biomaterials, drug development, nanomaterials, nano imaging, and drug delivery, theranos. And recently we have added new dimension, artificial intelligence also into this. So all these kind of things we need, because only then we can reach in a suitable solution to eradicate cancer from our body. When we are working on cancer, it is not only related to cancer. Actually, the targeting materials, which we are utilizing, that we can change to some other diseases, that also can be utilized, and we can deliver the drugs and change the drugs. So that is not a big issue. But particularly we are mentioning to cancer, it doesn't mean that we are working only on cancer. We are working on different diseases also. So only thing is that according to the disease and other kind of things, we have to change all these targeting or the drugs which we are using, the polymer which we are using, and many other materials we have to design. That's all. So in that way, when we are combining these things together, we can deliver the drug in a much better fashion. And for that one, actually, we have developed a lot of quantum dots which we can use in a different spectrum, and also some silica nanoparticles which we are utilized for to deliver to find out as a toxicity which we have given to a zebrafish, and we found that entire zebrafish is actually glowing with the fluorescence image which we have put some fluorescent materials in the silica nanoparticles. So that means all these materials is highly biocompatible and we can utilize for our work. And further, we have developed very good second NIR window deep tissue imaging materials which we have developed. So everyone this is our current first biological window region is there, and second NIR region is there. So there we can get a very good imaging because it will not interfere our biological fluids. So we have developed that kind of material so for a very good imaging. And sometimes we have to utilize some sensors to find out the targeting materials and other kind of things. So for that one we have utilized some selective placement for geo using LB methods and other kind of things. So and also we have developed a fluorinated graphene oxide for actually this one is actually really good for making as a drug carrier as well as magnetically responsive for imaging and photothermal therapy and photoacoustic contrast. We can use this one for one material itself. We can utilize it for imaging and delivering the drug, and even that itself can be utilized as a killing agent. So in that way, we can utilize this theranostics in a much better fashion. So, and also we have utilized developed materials for ocular imaging also. So that also we have used for gold nanoparticles for the development. So in this one, actually, I would like to focus four works which we have conducted, and I will go a little more quickly into this one, and I would like to introduce what are the things which we have conducted under this one. And some of the slides maybe I will be skipping with fast, but I have put this one in the PPT itself so that if someone is wanted some explanation, I will come back. I will come to that slide later. So okay, so though we have worked in many areas, I would like to mention in this talk only about the theranostics material which we have developed to kill the cancer cells for different cancer cells. So, and that actually first we have developed a triple targeted dual drug silica nano formulations, and then we have developed a lot of cobalt based nano quantum dots, but cobalt is actually a bit toxic, so we have changed that one to cobalt has been replaced with copper. So we have used the copper based nanocrystals for cancer theranostics. Then we did with the gold nano cages, we have used the combined nanotherapy chemo immuno using gold nano cages. And then actually we have used hybrid solid lipid nanoparticles against a brain tumor. So these are the four different things I would like to mention during this talk. So triple targeted dual drug silica nano formulations. So for this one, actually, we have developed a silica nanoparticles, and the drug which we have utilized the paclitaxel and suramin into this one. And we have covered this one with the polyethylene glycol PEG coating which we have given, because we found that whenever we are putting this into the silica nanoparticles, the drugs actually leaching out very fast. So what we did, we have coated with the polyethylene glycol for making it highly biocompatible little more. And then we have used 3 targeting modules, because then it will be, the targeting will be really good to the cancer cells. For that one, we have used CD31 and folate receptor and TEM, which we have used for these kind of targeting. And we found that whenever there is a non-targeting silica nanoparticle we have used, it is actually not at all targeting the cancer cells. But when we have put one each, and you can see that it has been increased the targeting ability. The drugs has been going and attaching to the cancer cells more. So by utilizing this, that we could kill the cancer cells more. So here you can see very clearly that when we are using the MCF7 and HUVEC and L929, these are the cell lines which we have utilized. So when we have utilized this drug, we found that on the third treated day and control and the treated, we have found that this entire things has been, this actin filament has been demolished by using this drug. And when we have used this one to find out in the HUVEC cell lines, MCF7 lines, MCF7, and we found that this one L929 is actually not at all having any issues, but all these other two cell lines actually killed. So that means our killing strategy is really good. And when we have used this one with HUVEC cell lines, you can see that this angiogenesis has been completely destroyed. That clearly means that we can kill the cancer cells by using the drug, as well as by using the drug which we can kill this angiogenesis also. So in that way, in both ways, we can kill the cancer cells. That is the advantage of using this drug. So then just I would like to show that when we are using the non-targeted cancer imaging, when we have used the non-targeted one, you can see that this entire drug has been gone to the liver side. So that is quite normal. So but when we have utilized our targeting, so then we can see that it has been gone to the correct where the cancer cell is there, and it has been started targeting into that. So then we found very clearly that when we are using, it is actually going almost all to the particular cancer area, and we can kill the, sorry, this one is not working. Okay, so we found that most of the drug is actually going directly into the cancer cells. So which resulted, actually you can see very clearly that when we are using the control, and from the control when we are using the triple targeting, we can see very clearly that the tumor mass has been heavily decreased. That clearly means that this dual drug and triple targeting method is highly effective, and it can be utilized to kill the cancer cells. So then as we have found that all these things we have to utilize the drugs, two drugs, and imaging moieties, and many things we have to put inside, and we have to work on that, and we found that it is actually a cumbersome work. So later for solving that kind of things, we have started working on copper based nanocrystals, in which we are thinking that imaging is also possible, and because of the NIR laser heating, we can kill the cancer cell also. So that means we need to develop only one material. There is no need to take silica nanoparticles, put the drug, and making the coating on polyethylene glycol. Then we have to utilize the targeting moieties to attach. So lot of works are there. So I think we have to reduce all the things by using only one or two materials, so that it can be highly effective and it will not be a cumbersome. So for that one, which we have created a copper based nanocrystals for cancer theranostics. So in that case, that we have developed a copper based material for this multimodal imaging, and for that we have developed a very good material which can be utilized with optical imaging as CT and photoacoustics, so that this material can be utilized for many imaging modalities. That is the beauty of this material. And along with that, we can also use this one for killing the cancer cells too. So here actually what we can see very clearly, this PEG coated copper sulfide we have utilized. Then we can see very clearly when we have used NIR laser irradiation, and this has been killed only at selected area. So you can see that here we have used the copper sulfide nanoparticles. So when we have used the NIR, you can see that very selectively it has been killed the cancer cells, but other areas has no issues. So that means after giving these nanoparticles into the cancer area, and after imaging it, we can see very clearly that okay, it has been reached there, and then we can use the NIR. Then in that way we can selectively target, and we can kill the cancer cells at that particular zone. That's the advantage of this kind of work. And Again, what we have used for a peg folate to, and also we have used the dogs also into this, so that we can actively increase the payload, the drug into that, and we can kill more cancer cells. So in that way also we have achieved a very good result into that. So then we found the material actually going more into the liver, kidney and spleen and brain, heart and lung. So here, it is not at all going all these areas, but we found that even if it is going into these things, it is not creating any issues. That means there is no toxicity based on all these materials, and it has been totally eliminated from our body. We can be eliminated from our body. So there will not be any issue based on toxicity of these materials, but this can be utilized for killing the cancer cells also. That is the advantage of this material. Then again, actually we have developed, when we found that okay, copper sulfide is really good for making, then we have added some other modification into that to make a little more higher level of material, so that in this case we can go to the second level of window for imaging. That is what we have interested to develop the materials. So based on that one, we have used a new material, and we have developed a new material, and we found that it is actually working well, and it can be killed the cancer cells, as well as can be utilized for different imaging modalities. And okay, this one actually very clearly we can see that it has been injected into the cancer area, and we have used the NIR for the treatment, and we found that very clearly that when we have used the saline and these kind of things, and there is no change into that. But when we have used the NIR, when with this material which we have developed, we found that this cancer can be killed, these cancer cells can be killed, and the size of the cancer is actually shrinked. So it found that it is really a very good work which we have conducted to kill the cancer cells by using non-toxic materials. And we found that after treatment, the control mass, the weight of the body weight of the mouse is actually increasing when the treated and the control mouse in the same fashion. That clearly means that it is actually not at all giving any toxicity to the body, and this one is actually eliminated within 2 weeks of time. Everything has been eliminated in our body after the treatment. So this one is actually a very good thing for the use of killing the cancer cells in our body. And these are some other materials also we have developed based on this one, and similar work which we have conducted. We found all these materials are really good to kill the cancer cells. Then based on all these things, we have tried to, we found that some of the times some of the pathways are not actually acting to kill the cancer cells. So what we have used type 1 and type 2 type of killings of cancer cells by using two different methodologies to kill the cancer cells. So for that one, actually we have developed gold nano cages to put the drugs inside the materials inside the gold nano cages, and utilize the NIR treatment to kill the cancer cells. So for that, actually we have used the combinational nanotherapy in this case. And for this combinational therapy, what we have developed dual targeting material. Actually, we have used the gold nano cages has been used for a biocompatible heat responsive nanoparticles for targeted drug delivery and imaging actually into that. And we have used the hydroxythymoxyphene, which we have used that can be worked against the breast cancers and positive ER receptors, which is really good. And we have used the Moran coating on the surface of the gold nano cages so that it can be contain this drug which we are putting inside the nanoparticle can be capped into that gold nano cages itself. Then we have used targeting moieties also into this one to deliver our nanoparticles to the site. So these are the characterization things which we have conducted of our developed nano particles. Actually, we have utilized these silver nanocubes for developing our gold nano cages. So it's a different types of process which we are utilizing, and we found that with the help of microwave synthesis, we can develop a very good nanoparticles for making some cages. So maybe I don't know whether you will be able to see some holes here. You can see very clearly. So that, yeah, maybe here we have developed the holes here into the gold nano cages, and we can put the drugs through inside this one. So like that, we have developed very good nano cages using the gold nanoparticles. So these are the different characterization techniques which we have used to check the gold nano cages. So what we did actually, we have utilized these gold nano cages to put this as a drug carrier for chemotherapeutic agent. We have utilized as a hydroxytamoxifen and moran polysaccharides, which we have used the immunomodulating adjuvant polysaccharides and TROP2 antibody as an antibody to target this to the cancer cells. So you can see that. Maybe you can see very clearly that this one is actually that while we are using the NIR treatment, slowly the nano cages actually getting heated up, and the drugs is slowly oozing it out from these cages. That's what we have done into these materials. And what we did this one here, we have clearly did some induction of autophagy and lysosomal impairment for killing these cancer cells. So normally, as you all know that, there will be autophagy will be there whenever there is a foreign body is coming, or maybe some damages happening into that, and this will become autophagy, and lysosomal will be coupled together, and automatically it will be taken away from the cells. But what we did actually, that whenever we are using this one and we are giving a drug, the same thing can happen. So there will be a drug resistance can happen. So what we did, we have utilized this nanoparticle to impair that method, that mode of autophagy and lysosomal interaction, and by that, the material will not be come out, and this can be additional help to kill the cancer cells, along with the materials which we have utilized, and the drug can be killed the cancer cells. So both ways, type 1 and type 2 methods which we can utilize to kill the cancer cells. This is what we did in this case. And I think I can skip all these things. So here actually what we did here, these nanoparticles, what we did, it is actually coming into the cells, and after coming here, we can see very clearly that an induction of autophagy and mitochondrial perturbation is happening here. So if it is happening, and then normally all this hydroxytamoxifen is actually a very good work against the mitochondria, as well as this nanoparticle is going into the mitochondria. So in that case, the mitochondria will be dysfunctionalized, and it can go to the cell death. That is one cell death method is there. So in other ways, if it is there, the phagophore formation will be there, and there can be autophagosome will come, and it can be eliminated. This is like these things can be eliminated from the cells. But what we are using these gold nanoparticles, we are giving more load into that one, and in that way there will be a leakage of this lysosome, and there will not be any kind of combination will not be there, and that lysosomal body, that lysosomal compartment will be break out, and the material will come out, and it will be killing the cancer cells. So in both ways, we can see there will be a killing of the cancer cells. So for that one, we have utilized the study which we have conducted, the visualization of these nanoparticles going into the cancer cells. and it is going into the mitochondria. I think maybe some of the things which maybe you will be able to see this one. Okay, sorry. Yes. So the previous video which we have shown is actually showing that this nanoparticle is actually going inside the cells and going into the mitochondria, as well as there is a lysosomal and this impairment is actually happening place, and the cells are actually killing. This is what we have shown. So in this case also we have taken very sliced the TEM images of that one, and we have very clearly shown each and every slide how this one is entering and how this one is going and killing the impairing this mitochondria, and it is actually mitochondrial swelling with clear specks between the cristae, and this is actually making clear killing of the cancer cells. Naturally this will be happening. So this one you can also see very clearly that why this one is actually not autophagosome is actually not working, because whenever this phagophore is there, when it is coming to fuse with the lysosome, because of these nanoparticles, gold nanoparticles already loaded into that one, and which is actually will not be in a position to come into fuse with the autophagosome, then this will also be burst out. So in that way we have shown very clearly each and every step, how this one is forming and how this one is killing, and everything we have shown very clearly, so that this can be killed. And along with that we have very clearly showed how this autophagosome lysosome fusion is also happening. And that's a 3D reconstruction we have shown very clearly into this. And this one is actually happening within the 24 hours of time. And this autophagosome segmentation also we have done, and we have very clearly shown that how this one is killing and entering into the cell, as well as entering into the mitochondria. And this clearly shows how this lysosome impairment is also happening. And this one you can see very clearly that TAN is actually binding to subcellular entities, and how this cell nanoparticles are going inside. All these things you can see here. Okay, these are the nanoparticles, gold nanoparticles, and these are the subcellular yes, you can see very clearly that this one is going inside everywhere and it is killing. And here we have used the FIB SEM for the treated MCF cell lines. So this one is also very clearly showing the degradation of mitochondria and alteration of mitochondrial membrane potential, and autophagy degrading the mitochondria and apoptosis lines based on these things. And this one actually we have shown how this nanoparticle is actually going into the mitochondria, and how this mitochondria is actually impaired. And you can see that some of these areas very clearly, if you are looking, I think maybe we have these areas, we can see that this nanoparticle is actually going and entering into that, and these are the areas where this nanoparticle is going inside and killing the mitochondria as well as the cancer cells. And because of that, TAN is accumulating. TAN means the gold nanoparticle which we are using is accumulating into the mitochondria, and this is creating the problem. This and it is actually killed. So we have also used the different methods, cell cycles and analysis for the necrosis. and that one is actually going for killing the necrosis is also happening, as well as lysosomal killing is also happening. So all these things very clearly shows that there we can use these nano cages for the theragnostics, imaging, as well as the killing of the nanoparticles together. So then we have developed a new material, is actually hybrid solid lipid nanoparticle against the brain tumor. So here we have developed a nanoparticle which we have utilized to kill or selectively the blood brain. It can cross the blood brain barrier, and it can be selectively kill the brain tumor. So we have studied this one, and the biodistribution of the material which we have developed. You can see very clearly that in the initial case, it is actually going to the liver. Of course, biodistribution is there, more it is going, and then it is going into the brain too. So what we have developed in this case, very clearly, almost all these, when we are using the targeting using the RGD and TF, very clearly that it is actually going into the cancer cells and not into the any other areas. So very clearly you can see when we have utilized this one is targeting to the brain tumor, and you can see very clearly that it is a dual drug which we are utilized. The drug is more going into the brain tumor. And this which we have used this ICP-MS study, then we have found that it is actually normally going into the cancer cells more, and not it is going into the other areas in our body. And if there are some materials are going in that, but slowly it is actually going out of the body, it is not creating any issues, it is going only to the cancer cells more. Then we found very clearly that you can see very clearly that is a dual drug is there. It is most of the things are actually going into the brain tumor. And when we have studied its behavior analysis of the mouse, then we found that the behavior of this mouse after creating the brain tumor, then we started injecting the drug into the brain, into the body, and we found that after some time, the mouse is actually showing is really a very good movement and all the kind of things. There is no problem for the behavior for that particular mouse. So that means this one is really a good material that we have developed so that it can be eliminated the brain tumor from the body. So you can see very clearly that this when we are using the double targeted nano material, this particular brain, we have found the therapeutic outcome is like this. So when we have used the control, you can see very clearly that that this one is increasing and it's not at all going down. But we found that whenever we are using one single targeting methods, still it is actually decreasing compared to the control. But when we are using the dual, we found that it is totally taking away the cancer from the brain. It's really a marvelous outcome which we have developed based on our solid lipid nanoparticles. So here you can see that when we have studied its survival rate, we found that more than 60% of the mouse has been survived while we are using the dual targeted medicine which we have used to kill the cancer cells in the brain. So that very clearly means that these methods are really effective, and we can utilize this material for our human beings in near future. And we have got the patent for this kind of this material also, and we found that this one is mainly good and we can be eliminate almost all the cancer from our body. So in that way, which I would like to mention that this theranostics is going to play a major role against the cancer therapy, because the current treatment methods are actually in a difficult situation to prove, so that to clear all the cancer cells from our body. These kind of methods, with a diagnostics approach, we can remove even almost cancer cell, even if it is not full, maybe 95% of the cancer cells can be eliminated from our body. So that is why this diagnostics approach is really good in the near future. And along with, actually, I would like to mention some of the work which we are working nowadays with artificial intelligence. So normally the medical imaging, nowadays is actually showing only the qualitative imaging. So with artificial intelligence, we can found the actual in-depth imaging also. That is what we are interested to work with artificial intelligence, and we have started working on that. So most of the things, nowadays, the analysis is also making some mistakes. So this artificial intelligence, when we are working with the medical diagnosis, can be an error free, and that would be a major revolution in the field of medical science. So in that way, what we are trying to develop a human on a chip program, and a human on a chip, so that we can develop the chip as a human chip, and can be directly connected to the artificial intelligence, so that we can analyze all the data and other kind of things, so that we can find out what can happen to this person even in the early stage itself. We can find it out. And with AI, that's we can create a medical horoscope. That's what we are trying to make, so that when we are analyzing it, we can find out the person will have some problem in within 10 years or 15 years or something, or current problems can be cured with using some of the other drugs or something like that. So all these things will be going to happen in the field of medical science in near future, with artificial intelligence and this theranostics and this nanotherapy works. It can be a marvelous thing for the new medical sciences. So by this, actually, I would like to stop my presentation, and these are my group members, which I would like to thank for all of their support and their work towards making all these materials for developing these nanomaterials in workable conditions. Thank you very much, and I hope I have presented in a nice way. But always, when talking to the computer, is always not in a good way to deliver the things which we are not feeling, we are not getting the response from the audience. So that will be there, but something is better than nothing. So I am open to the questions.
So there are a few questions, Dr. Shakti Kumar. So
yeah, yes, please.
Yeah, we can read it from here, right?
Yeah, yes.
Please.
So there are a few queries. The first one is, can you explain the use of liposomes in theranostics?
Pardon?
I repeat, could you explain the use of... So the question is, could you explain the use of liposomes in theranostics?
Yeah, liposome is actually, I have already mentioned, liposome is only a one mode of delivering the materials. We can utilize the many other materials also. So liposome is actually crossing the blood-brain barrier. We found that one is much better in the case of blood-brain barrier. That is the reason which we have utilized the liposome for that particular work. We can utilize, even sometimes carbon nanotubes. We found that it is very easy to cross the blood-brain barrier. We have utilized that one also, but I have not shown that that result here. That's all.
Okay, sir. Next question is from Vijay Kumar. The question is, what is the advantage of Mauran polysaccharide compared with other polysaccharides?
Yes. Moran is actually a good material which can be utilized. We can use attach some antibodies into that, as well as it can provide some immunomodulation into that. That's why we have developed this Moran into that one.
Okay, sir. Next question is from Archana Kumari. The question is, embolization is also an emerging therapy for cancer. So, can we use this technique to be compared to give a form of diagnostics?
Could you please once again?
She's asking whether embolization, a new emerging therapy for cancer, can be used in combination as diagnostic technique. Embolization.
I haven't seen such kind of things, so yeah, I cannot answer that one for time being.
Okay, sir. Next question is from Professor G.M. Nair. The question is, are there any specific receptors on cancer cell surface to which the nanoparticles are targeted? And can we make an early diagnosis of cancer using this technique?
Yeah, actually, we have utilized folate receptors and some of the RGD receptors, that kind of things which we have utilized for targeting the cancer cells. But a very recent paper came that single strand RNA is actually siRNA is also utilizing to detect the cancers in early stage itself. So I think if we can combine that kind of things with these nanoparticles, of course we can utilize to find out the early detection can be possible. So that is one of our areas which we are going to work. As I have already shown, these artificial intelligence and other kind of things which we are interested to utilize in coming our future work. We are interested to utilize these kind of things to early detection. So here we are going to use that one, and of course it will be highly useful for finding it out.
Okay, sir. Due to time constraints, we are winding up this session. Once again, thank you, sir, for the insightful lecture on diagnostics, the 21st century cancer treatment. Your lecture was highly enlightening and thought-provoking. Thank you, sir, again. And now we will move on to the next session. And
thank you very much. Thank you.
Thank you, Dr. Shakti Kumar. Thank you so much.
Thank you. Bye.
Okay.
now we invite.
Wait. Yeah, we are waiting for it. Okay. Dr. Jasmine, wait for a few minutes. Okay, we are waiting for the speaker. Wait.
Yeah.
Hello, can you all hear me?
Yeah.
Hello.
Welcome.
Hello, yeah, hi everyone.
Okay, just wait. Okay, you can. Just 1 minute. Yes, sir.
Now we invite Dr. Jasmine Shah from Department of Plant Sciences, Central University of Kerala to invite the speaker of this session. Dr. Jasmine Shah has received many prestigious fellowships like EST Inspire Faculty Fellowship, SERV Fast Track Young Scientist Fellowship, DBT Research Associateship, as well as IIT Postdoctoral Fellowships. Currently, she is involved in research on plant agrobacterium interactions, stress-induced mutations, plant functional genomics, as well as epigenetics. On behalf, we welcome Dr. Jasmine Shah to introduce and invite our next speaker. Over to you, ma'am.
Thank you for giving me an opportunity to introduce Dr. Neetu Singh, Associate Professor, IIT Delhi. Neetu Singh obtained her bachelor's and master's degree from University of Mumbai and her doctoral degree in chemistry from Georgia Institute of Technology, Atlanta. She obtained her postdoctoral training at the Harvard MIT Division of Health Sciences and Technology, US. Dr. Singh returned to India in 2012 and joined as faculty in the Polymer Sciences and Engineering Division at the National Chemical Laboratory, Pune. Later, in 2014, Dr. Singh joined as an assistant professor in the Center for Biomedical Engineering, IIT Delhi. In 2012, her important scientific contribution in developing strategies for managing cancer got her the Kiran Shah Mazumdar International Oncology Fellowship for establishing cancer-based research ties with MIT, USA. She is also the recipient of Innovative Young Biotechnologist Award 2013 by DBT India. In 2019, she received the Veena Arora Young Researcher Award by IIT Delhi, which recognizes best research contributions at IIT Delhi by faculty below 40 years. She is also the recipient of Nasi Scopus Young Scientist Award for Women in Science. She is also a mentor for an IIT Delhi startup, Kriya Labs, that is bringing valorization to agro-based biotechnological solutions. In her current program at IIT Delhi, she is establishing a research program that explores systematic probing into nanomaterials, biological activity, and formulates design rules for developing biosystems for specific biomedical and technological applications. Her group at IIT Delhi is interested in integrating concepts and skills for chemistry, material science, and biology to design nanostructured materials with enhanced functionality for applications in biomedical implants, cancer diagnosis, and drug discovery. So today her topic of presentation is new strategies for mitigating nanomaterial toxicity and enhancing bioactivity. So on behalf of the organizing committee, I welcome Dr. Neetu Singh for the presentation. Over to Dr. Neetu Singh.
Thank you so much for this wonderful introduction. And so I'm going to start sharing the screen. And I would like to thank Professor Mohanan for inviting me and giving me this opportunity to interact with all of you. So I'll just share my slides, screen share. Can you all see the slides now?
Just wait. Yeah, now it's possible.
Okay. Yeah. So would you like me to continue with the video, or because of bandwidth, I can switch off the video?
No, you can. You can. No issue. It's a high-speed band.
Okay. Okay. Let me just try to go back to the Zoom window. All right. So
you can use your madam. You can use your video.
I can use a video. Okay. All right. Okay. Fine. All right. So, the title of my talk today is New Strategies for Mitigating Nanomaterials Toxicity and Enhancing its Bioactivity. And, you heard from the introduction that I'm coming here. I belong to the Center for Biomedical Engineering at IIT Delhi. And, in our lab, what we have, okay, this is not moving. Yeah. So, before we get into the talk, I would like to tell you some research efforts, give you an overview of what we do here at IIT Delhi. And, in my lab, we are trying to address some very simple questions. For example, you all are aware that we can get tumor biopsies quite easily, and the tumor biopsies can result into, tumor cells. So the question we are asking is, can we utilize these cells to understand and intervene cancer? So um, basically, the other question, so this is part of our biosensing work that we're doing. We are also trying to address, issues of tissue engineering. So for example, we are trying to, if you look at this picture that I'm showing here, this is a picture of your knee region, so what you have, or any bone you can call it. So what you have in the bone is you have the bony structure and you have the cartilage structure, right? These two are very different in properties. So what we are trying to do understand is, how does nature mimic this alignment of different types of cells in different regions? How do you get the spatial control over where the cells go and how they grow and things like that? So this will be important because we can then come up with better tissue engineering strategies where we can get, have multiple regions of different types of cells in one scaffold. The third thing that we're trying to do is develop new strategies for drug delivery. And here we are trying to understand if we can use a new material like the RBCs. And I'll tell you today in the talk why we are motivated to use RBCs. We are trying to see if RBCs can be used to develop synthetic nanomaterials, which can take up more hydrophilic and hydrophobic drugs. Because to me, RBCs are the natural carriers, right? So they're not in nano, but they are micro carriers that our body has. So why can't we utilize the strategy that nature uses to deliver things or to carry things, and extrapolate that into nano regime and then come up with a better strategy? So that's the whole idea that we are trying to work with. Okay, so now to introduce you to the research field in nanomedicine, usually what you see is you have, there are two approaches. One is the bottom-up approach where you have, you design a system and use it for various applications. And that's what I've told you. We are also utilizing nanoparticles in all the applications that I was talking about. So for example, the biosensing work, we are trying to come up with a nanoparticle which can be used in biosensing. We are trying to develop nanoparticle-based strategies for tissue engineering and even for the drug delivery with RBCs. So, typically in the field of nanomedicine, this is called as the bottom-up approach, where you design a system and use it for various applications. Now, what are the type of nanoparticles? I'm sure the audience are aware of different types, but just for a recap, you must have heard of liposomes, you must have heard of quantum dots, gold nanoshells are, they're used for hypothermia and a lot of other cancer therapies. Mesoporous materials like mesoporous silica particles, we have micellar structures, magnetic nanoparticles, dendritic polymers, or carbon nanotubes. So all of this basically a different class of nanoparticles which are used for various applications. These are, of course, decorated with multiple types of polymers. For example, polyethylene glycol is typically used so that you reduce non-specific protein adsorption. And I'll tell you in a little bit what is the importance of the non-specific protein adsorption. You can have, a core shell kind of an architecture where you have a core with a different material. So, for example, in quantum dots, you have CDSE core and a zinc sulfide shell, which gives that beautiful fluorescent properties to these materials. You can also put in folate. So you must have heard of targeted therapy. So, there is a very common group which is used which is folate, because the cells have a folate receptor. So the idea is that if you have folate on your surfaces, the particles can go and attach to those cells better. So these are very commonly used strategies that all of you are aware of. And of course, you have to make, with these strategies, you have to, and the options that you have, what the field has been doing is trying to make a multifunctional nanoparticles. So we become a little ambitious as a synthetic chemist, and we want to incorporate all the properties that you can think of. For example, you want to use these materials for imaging. You're going to use magnetic particles. You will also put in liposomes that you can encapsulate. You will use electrostatics. You will have layer by layer assembly. So you want to put in imaging. You want to put in therapy. So there is a concept of theranostic that people talk about. You want to make it targeted, so then you will have surface receptors and so on. So it's a very complicated system that we are trying to develop in nanomedicine. Unfortunately, with all these different strategies, we have been very unsuccessful in, just a second. This is blocking my presentation view, so I'm going to move it in this side. Yeah. So, it is basically, we've not seen translation into the clinics. All the strategies that I just discussed about, we have used all of that, but very few nanoparticle-based strategies are present in translational in clinics, right? So clearly, there is a need for a new approach. Maybe this is not the approach we should be taking. We should be doing something totally differently. And we will discuss sometime understanding how you can change things and how we can improve the bioactivity of these materials. And of course, you need, so you need to develop more top-down approaches, where what we do is we first understand the interaction of these systems with the biological entity. So you understand the biological interaction, and then with this insight, you need to develop a new system. You need to design a new system. So the solution probably is get more insight on the cellular interactions of these samples, right? So basically, again, here what I'm proposing is that instead of making a new nanoparticle, we try to understand the interactions that these materials have with the extracellular space and the intracellular space. So you do that, then you design your material, you characterize them. After the characterization, your characterization is going to involve understanding this extracellular and intracellular interactions. With this understanding, you start to iterate. You start to iterate your nanoparticle and try to understand. This becomes a cycle where you're doing synthesis and you're characterizing them well, going back and studying your interactions, and then doing a lot of multiple iterations. And ultimately, you might have something which can basically get you the nanoparticle for drug delivery or imaging or any application that you want to do, right? So just to spend a little bit of time on what we understand on toxicity of nanoparticles, and because I'm going to spend a lot of time talking about our strategies of mitigating what we've done in the lab for mitigating toxicity first, and of course, then developing applications, right? So if you look up the literature, you will find that the size of a nanoparticle is extremely important. And there is some consensus among people among the scientists that anything which is, so for example, what is small or what is big, right? So anything which is smaller than, say, 5 nanometers is too small, and it has high probability of inducing cytotoxic effects. The reason being, you can imagine them interacting with the proteins or intracellular proteins or extracellular proteins more, and that will disrupt the folding patterns, right? And that is going to cause toxicity. So, then what is the ideal size? So, maybe it has been shown that by many reports, it has been shown that, an ideal size could be between 10 to 30 nanometers, which can be a best compromise between uptake and toxicity, So, that's, sorry about this noise. We have an airport close by, so that is why you'll keep hearing this every 11 minutes. So it has passed now. Yeah, so you will have, there could be a best compromise between the uptake and the toxicity, because if you make it really big, you will probably have low toxicity, but because of interactions with the protein and intracellular components, but you might have toxicity because of uptake or other reasons, right? So again, if you have greater than 40 nanometers, the cellular uptake decreases. It has been shown that the cellular uptake is decreased, and maybe very little gets inside, and then you cause off-target effects and other things, because it's lying around everywhere. So, so as I said, maybe happy compromise would be between 10 to 30 nanometers. There is a lot of reports about the shape of the material. For example, it has been shown that spherical materials have highest uptake, but also highest toxicity. There is arguments where people say that they have rod shape, which diminishes the uptake, but then there is less toxicity. But then there was a report a few years ago which showed that the anisotropy actually causes toxicity more, because this can penetrate through, and it can actually acts like a needle and gets inside the cell easily and ruptures the cell membrane. Okay. Purity of the material is very important. For example, we are working with metal oxides, and it's important that these metal oxides do not have any free or other material which can leach out, which can cause toxicity. These are heavy metal, and they're very toxic for the body. So that's why these considerations are given. Of course, there are strategies where you put coatings around them so that the leaching is reduced and things like that. Nanoparticle stability is very important for toxicity. For example, acid degraded cores of iron oxide or quantum dots can actually, they can degrade the material faster, and you will release the if it's a polymeric material, let's say, you will release the polymer fragments or the metal oxide surfaces even more faster, and that can cause more toxicity. Again, coating the nanoparticle has been a solution for that. Nanoparticle surface charge is extremely important, So highly positively charged nanoparticle have greater tendency to interact with biological components, and it can actually cause more toxicity compared to, slightly less positive charge. And what is more and less depends on the system you use, but usually 30 millivolts of, the potential is what is considered as the borderline. So surface functionalization, you can alter the nanoparticle properties. For example, you can coat them with, a polyethylene glycol or something else, and then you can address these issues. So overall, if you want to understand toxicity, in vitro systems, one of the key parameters is the Ross generation. So it is understood that if the nanoparticle generate more of reactive oxygen species, so by the way, there are Ross generation happening in our system already, okay? It's part of the biological process. It's part of how the cells regulate the environment. And, it causes oxidative, of course, if you generate more Ross, because of some external factors, for example, nanoparticle in this case, you will create oxidative stress. And that actually is known to, cause cell death and DNA damage and cell cycle arrest and so on. It can interact with the nucleus, the nucleic acids in the nucleus, and cause a lot of toxic effects, right? So Ross generation is considered as one of the key parameters. Interestingly, because the surface of a nanoparticle is lot of oxides, especially in inorganic material, and it also gives a lot of surface area. There is a catalytic property, there's catalysis of Ross generation. So, and we will learn about it later in the presentation. Yes. So are we taking questions now? Because I see raised hands, or can we take questions at the end?
Can, ma'am, we'll try at the end.
Yeah, we'll take the questions at the end.
Yeah, we're following like
Okay.
that.
All right. Yeah. Okay, so the second part, of nanoparticle toxicity is that the nanoparticle can actually, the way it interacts with the cellular membrane, does it get endocytosed? Does it enter the lysosome? How long does it stay? Because longer it stays in the endosome or the lysosome, it activates the toll-like receptor proteins, and that actually signals the cells to starts the apoptosis process. So that's considered how long, I said if it stays longer, what is long and what is short. So those kind of interactions are very important when you're trying to address the toxicity of your nanoparticle, right? If you consider, so once you study this in vitro, then you move on in vivo. And in vivo, typically what we're looking at is what is the pharmacokinetics of these nanoparticles, what is the metabolic toxicity like, where does it get absorbed, how is the distribution, what is the excretion pattern like, is it compatible with the blood, so what is the hematological toxicity, is it heme compatible, what is the immune system toxicity, does it activate certain interleukins or cytokines, typically we study that. Ultimately, what is the genotoxicity like, does it affect the genetic makeup of the organs or genetic makeup of the cells which are present in those organs or tissues. You look at specific organ toxicity as well. So what happens to the lung, the liver, the heart, and things like that. So this is basically, you can have effects of nanoparticle toxicity in all these aspects, right? So if I want to make a nanoparticle, so I hope by now you've understood that the key aspects, if we want to mitigate cytotoxicity, has to be the intracellular interactions. We need to understand the intracellular interactions and we need to address that. So if I want to mitigate the toxicity and improve the biological activity, if I play around with the intracellular interactions, I can do that. I'll have to probably address the protein corona formation, how long. So for example, a particle which has longer, it circulates in the system longer, just like your blood cells, maybe it can have more toxicity or it can have reduced toxicity, because if you change the protein corona formation on the surface, you're going to change the entire pharmacokinetic properties, right? So that's one thing that needs to be considered. The third very important aspect is the ROS generation. How well is the ROS generation on the surface? Is it generating, is there a catalytic effect or what is happening here, right? So these three becomes the key aspects that you need to address when you want to mitigate cytotoxicity and enhance the bioactivity. So I'm going to move to the first one and try to tell you how we have addressed that. And let's talk about the intracellular interactions. So nanoparticle properties that can influence cellular interactions can be size. I just explained to you, and there are a lot of reports which talks about how the uptake is dependent on the size of the particle. For example, gold nanoparticles had been a model system where they have studied this. How cellular uptake kinetics is affected, if there is a different size. Of course, more particles getting inside for a long, for a particular time, it will cause toxicity. So you can vary the size to reduce that. How shape has been affecting that, and we have studied that. How mechanical properties have affected. So, for example, the uptake mechanism is very, very dependent on the mechanical properties, and we will talk about it again a little bit more detail in some time. Surface charge, and I'm sure you all are aware of these properties of the material which affects intracellular interactions. The surface lipophilicity, for example, how hydrophobic the surface is, how hydrophilic it is. In fact, in one report in Nature Materials in 2008, they have shown patterned surfaces, which, so you have more pattern, and they showed that there was more cellular uptake, which makes sense because remember, even the cell membrane is a slightly patchy surface, right? So maybe if you have a patchy nanoparticle surface, you can increase the uptake and maybe decrease the toxicity instead of keeping the size the same or something of that sort. Here also they have shown that, what was the so you change the functional groups on the surface and you change the cellular uptake. In fact, you can make it enter the cancer cells more versus normal cells and things like that. So a lot of time has been spent in understanding the mechanism of uptake as well. Okay. So we actually were very intrigued by that. And, there was another paper very recently, I think this is in 2012, from a group at Harvard where they talked about, they had a simple nanoparticle and they said that to change the elasticity of the nanoparticle, to keep the sizes the same. So remember, I told you that the size range that you get to modulate is between 10 to 40 nanometers, right? Because that's considered as the ideal size if you want to reduce toxicity, increase uptake, and things like that. So, if you want to keep the sizes same, how do you change the uptake of the nanoparticle? So here they were talking about keeping the sizes the same, but playing around with their elasticity. So they had a nanoparticle which was 45 kilopascal, stiffness versus, a 19 megapascal, 1.6 megapascal. And, this is basically what they showed was that softer the material, or maybe an intermediate, not very soft, not very hard, you have more uptake, right? The argument that they gave was this. So they said that the cell internalization pathway shifts by varying the particle elasticity. So soft enters the cell via two pathways. So if you have a soft particle, it can enter the cell via two pathways. One is fusion, which is a predominant feature here. The other is endocytosis, which is lower. Now, remember I told you that if it remains in the endosome, it activates the TLR or the toll-like receptors. So what you've done here is by changing the elasticity, you've not only increased the uptake, you've also changed the toxicity pattern, so now your TLRs will not be activated, right? So whereas hard particles enters the cell only via one method, which is the clathrin-mediated endocytosis. So this is basically, again, a very interesting observation which they made. And we thought that, can we use these kind of insights and develop a material for some application? So the application we chose was RNA interference, because now we're talking about CRISPR-Cas9 and we're talking about gene editing. And what it involves is it's one of the modern tools of medicine, and it provides us, unprecedented opportunity to address a lot of diseases. Why? Because remember, 85% of the biological targets that we know of are non-druggable. Until the synthetic chemists come up with drug targets, we have to address these diseases if we know the biological targets by gene therapy, maybe. Okay. Now, what are the challenges? So I want you to focus on, the mechanism of the RNAi interference for a little bit. So, typically, the mechanism involves, you deliver an siRNA or this small interfering RNA. These are short sequences. These are about 18 to 25 nucleotide sequences. Usually, what happens is you deliver them as a antisense strand and the sense strand as a complex, as a duplex. It enters the cell and gets opened up. So you can also have a microRNA, by the way. So that's a different mechanism. But ultimately what happens is the microRNA or the siRNA opens up, and the antisense strand goes and interacts with something called as the RISC complex. Okay, so you have so this is the picture. This is the RISC complex, the red part, the green part, the purple part. The RISC complex is RNA-inducing silencing complexes. And so what it does is the antisense strand sits here, it starts to cleave, and that translates the information which is present in the antisense strand into the cytosol, and then ultimately the protein reduction or upregulation happens. That's how it controls it. So of course, nanoparticles are a strategy to address this, and you can easily utilize this. Now let's go back to the mechanism. So what did I tell you? I told you that, this is the RISC complex and this is the antisense strand, and you have the nanoparticle somewhere conjugated. So imagine, this is the RISC complex, you have the nanoparticle, and you have the sense strand. I hope I'm visible. And you have the antisense strand. This needs to interact with the RISC complex, needs to interact with the nanoparticle via the strand that you're putting in. Okay. So, so this is basically the picture. So this is the specific protein that in the RISC complex that we're looking at is EGO2, ergonaut protein, and that is involved in interaction with the antisense strand. So if you basically make it a very simple interaction, you have this kind of a picture. You have a nanoparticle with your siRNA. Of course, it's a duplex. You can also put a single strand, right? But that's not very stable. So that's why people use duplexes. And then this needs to interact with the EGO protein. And ultimately, you will have RNAi. Of course, remember, it has to enter the cell membrane. So those changes, there are a lot of studies which talks about how the nanoparticle enters. I am assuming that everything has happened, okay? So the question we asked in the lab is, can we get a systematic understanding of this correlation between gene silencing and the physical properties? I just told you that how the conjugation strategy, the size, the softness. So why did I say conjugation strategies? Imagine, so if this interaction will have some sort of a steric interaction, right? If you want to remove the sterics, the best part is to remove the siRNA. Once it enters, you can have a disulfide bond, and the disulfide bond can get cleaved by the glutathione in the intracellular space, and it can actually interact with the AGO2 without any steric interactions. That also means that you are compromising the stability of these materials, because once remember, a lot of thiolated proteins are present even in circulation, and once these materials are in circulation, they are going to release the siRNA even before it reaches the target, right? So there is this complicated processes that are multiple processes that are happening, which you have to address when you're developing a nanomaterial. It is going to cause toxicity because now the siRNA is going to get recognized by the body, and maybe your nucleases are going to start chewing it, and that can cause off-target effects. So that toxicity will be there. Of course, it is going to have low bioactivity because not enough is reaching the target, right? So if you want to address all of this, the best strategy is to use a conjugation strategy which does not allow the siRNA to come out of the nanoparticle before it reaches the target. And that is why people use different conjugation strategies, right? So, and elasticity, I just told you that elasticity. So think of a situation where you have the nanoparticle and you have a very soft material. Now, there won't be any sterics because the soft material is more malleable. It can change shape, right? So there won't be any sterics. So that was the hypothesis that we were talking about. And then can we use this to make better strategies? That was the question, of course. So we started looking at the steric effect and steric interactions and how do we alter the steric interaction. So this is basically the system that we developed. We developed the polymeric systems, which were small and large in size. So small were less than 800 nanometers and large were more than 100 nanometers. So large was typically around 200 nanometers. So we didn't go into more than 500. Okay, so it was around 200 to 150 nanometers, and small was smaller than that, 50 nanometers to 80 nanometers. The materials were the same size and same surface properties, but we had gold core inside them. So now they can't change shape. So they're not elastic enough. Okay. So there is a hard core inside them. And we did a simple study where we tried to look at how is the interaction of these multiple nanoparticles that we have made with the AGO protein. Okay. Very simple study. Of course, we characterized the material really well. We looked at it by TEM, tried to understand how thick the shell was, what the self-surface properties were like, how do we change the size of the particles, the charge of the particles, all of that, of course, were controls, and we had taken care of that. And of course, we looked at the hemocompatibility, the the cell compatibility, and the serum stability, and, was it able to protect the siRNA and things like that. So all the preliminary studies were done. Whether the cells can take up these materials, those were also studied. And very important, do we have any free siRNA? Because remember, in these structures, if I have free siRNA, I will still see effect, right? But I will not be able to understand whether it is because of the nanoparticle or the free siRNA. So we had to ensure that there were no free siRNA, and whatever siRNA we had was conjugated to the nanoparticle, okay? And of course, so we could characterize all of that, how much siRNA was loaded, was the siRNA loaded in the same concentration in all the particles, and so on. So then we did a simple experiment where we had the nanoparticle and we had the AGO protein. Our AGO protein was labeled, okay? So we had a fluorescently green protein and the nanoparticle was labeled red. So if they were interacting, you would see a yellow color. And this would tell whether they are co-localizing or not in vitro. This is not inside the cell. This is in vitro, whether they are interacting with each other or not, right? So we had to optimize this, and we basically made the model system to study these, to do these studies. Once we've done that, we tried to look at what sort of interaction? How do we quantify? I told you that I want to quantify the interaction, right? So in this experiment, we just showed that they're interacting because you have a yellow color. Red and green would give you that. But what is the level of interaction? Are they interacting? How do we quantify the interaction? Because that would tell us, right, whether they are interacting more, better, right? So we have to quantify. So we developed an in-house assay with ethidium bromide, which basically intercalates between a duplex, and more ethidium bromide would mean more interaction and things like that. Okay. So with this assay, we did our studies and we were looking at what happens with different nanoparticles. And what we saw was that a small soft nanoparticle has the greatest interaction. We expected that, right? It's small in size, less sterics, it's softer, even better. And then we expected that a small hard particle should have, because the size will play a bigger role. Unfortunately, what we found was that the soft larger particle had a better interaction. In fact, a soft large and a a soft large and a hard large particle did not have any significant difference. So if you look at the large sizes, the size actually had similar interactions. Size did not change the interactions at all, unlike what hypothesis I had presented to you. The huge difference we saw was between small hard and the small soft, which again this was kind of a surprising result, and we spent a lot of time trying to understand. And we did a lot of cryo EMs to look at where the proteins were interacting and sitting. And also basically we confirmed our so this was just the interaction with the assay. Does this interaction translate into the ability of these particles to silence the genes? We did that by RT-PCR, confirming by RT-PCR, and also by endpoint assays like the apoptosis. Ultimately, we were knocking down a particular gene like PLK1. It's a kinase gene, and we were trying to knock down that, and that has an effect for toxicity. So we were trying to look at apoptosis and how that affects. And our results were similar. Whatever we saw in the mechanism study were translated even in the RT-PCR and other studies, right? Which is again similar to what the literature also has. What we found was that, so then when we put our things together and try to come up with a mechanism, what we proposed was something like this. So if you have a size, in consideration, so you have a small nanoparticle. The small nanoparticle can easily interact with EGO2. If it is softer, the interaction is better. If it is harder, the interaction is slower. So we saw that there between soft versus hard in a small size, soft nanoparticles were the best, right? So this makes sense. There is efficient access to the siRNA, and here there is a difficult access to siRNA. In fact, it was so difficult because of the hard size that it was the lowest. We did not see very high in gene silencing with this. With a larger particle, you have a scenario like this. Instead of one interaction, you have multiple sites where they can sit and interact. And that is why a large, because it has more surface area, you have more surface and more proteins, more siRNA on the surface, more proteins can come and attach, right? So you get a flower-like arrangement here. And in cryo-EM, I don't have the pictures here, but we saw beautiful, nanoparticle with small protein attached there, right? So that's how we came up with this hypothesis. So ultimately, with this study, we were able to conclude that you should have a large and a hard nanoparticle, which will have better interaction. Small and soft nanoparticles, large and hard nanoparticles may result into steric hindrance and have less interactions, whereas small and soft will have better interactions, right? So with this kind of a strategy, we were able to develop vehicles which can deliver siRNA better and also reduce the toxicity. Right. So this is we did many other studies where we looked at the conjugation strategy, tether length, stand orientation, size, soft versus hard. So this is just a summary of all of that. I'm not talking about those things. You can read up our papers to understand them better. But basically what we did was we came up with some design rules for siRNA delivery, and we were quite happy about it. Right. So moving on to the next concept of addressing protein corona formation, right? So what we did here is, you understand that protein corona is very important. There are some disadvantages because it can cause unwanted immune response, but there are some advantages as well. I don't have to tell you why nanoparticles are used in drug delivery, but basically, what happens is the moment nanoparticles enters the system, enters the host, we're talking in vivo sense, non-specific protein adsorption happens on the surface, and that can actually hamper the bioactivity of the material, right? And that is the reason why people have been trying to put polyethylene glycol. People have used multiple strategies like polyethylene glycol or putting it with albumin, coating it with albumin and things like that. So if you look at the successful particles which are in clinic, Doxil is one successful particle, which is a liposomal drug delivery vehicle, which has polyethylene glycol on the surface, and Abraxane, which has albumin-bound paclitaxel, okay? But is that the complete solution for achieving the best drug delivery efficiency? Probably not, because pegylated nanoparticles are now known to cause anti-PEG antibodies. And there is also a concept which is complement activation related to pseudo-allergies, or CARPA effect, which happens because of enhanced pegylation, okay? So this basically becomes a problem. And that is why they have not seen a lot of success with pegylation. So, we turn to RBCs. We try to look at how, as I told you, RBCs are basically natural circulating particles, right? So basically, they are, discoid shaped. They are biconcave disc. The size is about 7.5 microns. And nothing sticks to them. They do not have non-specific protein adsorption. They are circulating cells, right? And the reason for that is considered to be it's called the stealth property. So if it doesn't attach non-specific protein adsorption, it's called stealth property. And the reason for that is understood to be the presence of CD47, which is basically activates clustering protein, which is a signal regulatory protein alpha, or SIRPA, which causes clustering of this and then binding, and then it signals the phagosomes that this is a self particle and they don't eat up, okay? So this is basically the concept is, if you take the cell membrane, it should have your CD47 and you coat it with the nanoparticle, you will probably get something which is similar to RBCs, right? Because the CD47 protein is important. People have tried coating the nanoparticles specifically with CD47. That has not reached to any conclusion. The reason being, remember, this is a protein which requires clustering, right? So the density of CD47 becomes very, very important, which we will not be able to mimic when we do synthetic strategies. So to avoid that, what they've done is they just take the cell membrane, they just take the RBC membrane and coat the nanoparticle with that. So you do that, but then loading of the nanoparticle, loading becomes an issue. The coating becomes an issue, and you still have a lot of other things which are present in the RBCs. So what we thought was, can we actually break the RBCs and form a liposome structure or a synthetic nanoparticle out of that, right? So to me, as a synthetic chemist, they are just a bag of, a bag which contains a lot of other proteins, which if I remove it, I can then get the raw bag and I can break them down and form nanoparticles out of that. So that's exactly what we tried in the lab. And we basically tried to look at, can we use these nanoerythrosomes as personalized medicine concept, right? So you can draw blood from the patient, create the nanoparticles, and then use it for the delivery of the nanoparticles, okay? So this is the RBC derived. What we do is we take the erythrosomes. I'm going to skip the technical part and how we synthesize it. But basically, the idea is you have all these nanoparticles with your proteins. You have your embedded drugs in them. And then can we deliver? So we optimized this process, and now we are able to create these materials for drug delivery. We have loaded dyes in it so that we can visualize it. A drug, which is camptothecin, which is a slightly hydrophobic drug. And the reason why we chose this drug is because, this also has an intrinsic fluorescence. So it allows us to study where the drug is present. Also, one of the less potent version of this drug, which is slightly hydrophilic, is used in the clinic. And camptothecin, which is more potent, is not used because it doesn't have solubility in water. Okay, so this becomes like a good drug to study. So I'm gonna skip all the characterization, and of course DLS zeta potential was studied. Thorough characterization of the cell membrane and what kind of proteins we have there. What is the stability like, because the whole concept was increasing the stability because they will reduce protein adsorption. So we did all of that. The biocompatibility was studied, like percent hemolysis was studied because this was derived from red blood cells. Cell viability and of course protein adsorption was studied. So you can see that they were very similar to the RBCs that are present. So the RBCs that we have, hosts are basically breaking down and resealing it again. So those are similar to RBC surfaces, and they were very similar to RBCs. There were no protein adsorption which were there. So our hypothesis was correct. They will basically not have any protein adsorption, non-specific protein adsorption. Now, because this is coming from blood, we were worried about the immune compatibility, and we started looking at the presence of TNF-alpha. We did see a little bit of immune response, but not much, not significant. It subsided down after longer period. So 12 hours, we did see a little bit of immune response, which subsided down, which is very typical of any nanoparticle that you deliver. You have initial immune response and it subsides down, right? So we were very happy with that. We started doing our drug loading and release studies. I'm going to skip all of that. I'm going to take you to the biodistribution. So because they were less protein adsorption and they were circulating longer, we were able to see really high amount of, so you can see that in blood, they were there even till 48 hours, 10% in 24 hours, which is quite high, you have to believe me. Most of the nanoparticles do not remain even in that concentration in the blood. And it was getting accumulated in the spleen, which is again, and the liver, which is again very typical of RBCs. So RBCs can get inside the spleen and the liver very easily, and that's where they get cleared, okay? So we developed better strategies for drug loading. I'm going to skip all of this and just show you that with this, we were able to actually show that they were there in the tumor for a longer period of time. And we saw improved, drug efficiency, improved delivery of the camptothecin, and improved survival of the animals when we did these experiments. So it was not, it was not a complete cure. We started to see slight increase in the tumor volume after 20 days. So we have not reached complete removal of the tumor because after 20 days, it started there. You can see the upward trend. So we are working on getting this better. But of course, this, you can see that the tumor was quite significantly reduced. So this is the tumor that we retrieved, right? So it was significantly reduced. Maybe we need to work with the dosing, maybe the dosing can be given again at day 15 where the tumor starts to grow, right? So we are working on better strategy for managing this. We're not there yet, but we have seen improvement in the bioactivity. So of course, the tumor volume was not reduced to the extent that we wanted, but we did see when we looked at the histopathological studies, we saw that there were slight improvements in the histopathology of these organs, So for example, compared to free and uncontrolled, we saw less metastasis, which is important because it actually, it is also adding value to the treatment that we're doing. So, again, for intestine, we saw similar level of toxicity, so it was not better. So we had a mixed result over here, but overall, we found that the animals were surviving better after 1 month. We are doing our longer term studies now. So how much time do I have left?
You can take 5 more minutes.
Okay, so I'm going to quickly wind this up in 5 minutes. So the third point that here, I hope I have convinced you that how you can change the size, the elasticity of the material and improve the bioactivity. You can also work with the protein corona and you can improve the bioactivity. And you can, the third part is addressing the ROS generation, right? So you all know that ROS generation, I told you that how toxic it can be. I just want you to pay some attention to some papers that, which published in 2012, 13, 14, and keep seeing these papers every day, which is about graphene quantum dots. So carbon, why did we choose carbon-based quantum dots? Because remember, they have beautiful fluorescent properties. They are not inorganic materials, so they are not very So we actually read these papers and we found that they're not toxic, right? So if you look at these data, addition of a 400 microgram of quantum dot decreases the cell viability 20%. And if you go in higher concentration, it actually decreases further. So you somewhere have conditioned that you can use your quantum dots only in this region. There was another report which talks about how these nanoparticles are biocompatible. But if you look at the cell viability, even with polyethylene glycol, 50% of the cells were dead. The report was looking at 50% cell viable, but for us, what we understood was that 50% of the cell was dead if you have 1 milligram per ml concentration, which is fairly low concentration, right? For drug delivery, you might require a little larger concentrations. So we thought that because it's a highly oxidative surface, it could cause toxicity. So this is Ross generation, So Ross generation can happen on the quantum dot surface because of a highly oxidative surface. Secondly, these are very small particles. These are tiny particles, 3 nanometers. The moment you increase the size of a quantum dot from 5 nanometers to 20 nanometers or 50 nanometers, you lose the fluorescent properties, right? So basically, I'm just showing you more data suggesting you have larger sizes of the carbon dots. They have less toxicity. So one strategy would be to increase the size, but we cannot increase the size here because you lose the property. So that is why I'm presenting this example to you, that there are certain ways where certain conditions where you cannot increase the size, although it would mean reducing the toxicity. So the problem we had was, how do we increase the size, alter the surface so that it generates less ROS while maintaining the photophysical properties? Simple answer to that for us was mask the surface. Have you seen this Lay's packets or the chips packings that we have? So you have small chips in a bigger package, right? So you have smaller quantum dots in a bigger package. That's what we envisioned. And what we did was we made something like this, where we create our carbon dots. And this was done with collaboration with Professor Pillai from Sikri and Vijay Mohanan Pillai. And he was kind enough to teach us the synthesis of carbon dots and a little more about quantum carbon dots. So basically, we created these carbon dots and we embedded them into a polyethylene glycol network, similar to, the Lay's picture that I told you. And we were able to maintain the fluorescent properties. I'm going to show you that what they look like. They look something like this. You have a 80 nanometer particle. So this is 100 nanometer scale. So this is because of the TEM, it has become elongated, but these are spherical particles in which you have carbon dots embedded. The photo optical properties were maintained. The fluorescence was maintained, and we did not see any diminishing of that. We started looking at Ross generation, and as our hypothesis was correct, now you have. So, in literature, people have tried coating the carbon dots with polyethylene glycol. And the first paper that I showed you here, in this one, this is basically that. They coated the carbon dots with polyethylene glycol, and they showed that they have improved the toxicity. But it doesn't work because the polymer coating is very little. So with our system, you have more polymer, so it can quench ROS better. So we showed that, indeed our hypothesis was correct. We looked at it, used this for intracellular imaging, we looked at how much intracellular ROS can it quench. And indeed, with the polyethylene glycol embedded quantum dots, we were able to see very little ROS production. So we used it for drug delivery and showed a lot of applications out of that. In fact, we have used these for developing sensors, biosensors for managing this, and that's a different talk. I can talk to you all later, or you can read our papers about it, where we looked at how these nanoparticles, these carbon dots, can be used to image the microenvironment in real time, because we can make these nanoparticles pH sensitive as well. So those are some applications that we are exploring now. So I hope I've convinced you that we have a strategy which helps in reducing toxicity of graphene carbon dots, because it produces low intracellular ROS and it renders multi-functionality to the carbon dots. Okay, so I'm going to skip my intracellular pH sensing part. and I'll come to acknowledgement. All of this work that I'm talking about would have not been possible without a generous funding from my home institute, IIT Delhi, and DBT, which funded my lab in the initial years, the IYBA scheme. DST Nano Mission funded the elasticity work, and I'm very grateful to them. They supported me when this seemed like a blue sky idea, when there were no literature papers about this, and there were not a lot of discussions happening in the field. So, they supported me when I proposed that, and I'm very grateful to DST Nano Mission for that. After that, after the first round of funding, DBT Nano Biotechnology Task Force have been supporting me and very generously, and they have supported the lab, and we've been able to do a lot of work. The RBC work was supported by them. Again, seemed like a very crazy idea. You're considering RBCs as a liposome. So that was an argument we had in the panel when I presented this proposal, but they supported me anyways, and it has resulted into some wonderful materials, and now we're exploring further with that. It cannot be done without all the hard work. I'm presenting this work to you, which is hard work of my students, the current and the past members of this lab. This is work of my 2 PhD students, so I'm thankful to them. And now I'll open to any questions. I'm open to questions. Yeah. Thank you. Thank you for patiently listening.
Thank you, ma'am, for the wonderful session on new strategies for mitigating nanomaterial toxicity and enhancing bioactivity. It was quite an interesting and enriching session. And now you can address the questions from the audience, ma'am. Actually, do I need to read it out for you, or you can?
Okay, so we have, I can open the question and answer. So thank you for the presentation. How do you avoid that RBCs do not take in the nanoparticle? RBCs do not take in the nanoparticle. Yeah. So the interaction with RBC. So, once we modify the RBC, the nanoparticle, once the nanoparticle has become, once we create the nanoparticle out of the RBCs don't eat up each other, right? So because of the CD47 probably, and that's what we looked at. We looked at that the CD47 is maintained on the nanoparticle as well. So we did not see any uptake with the nanoparticle. How do you ensure, because remember, we have not modified the cell membrane too much. We are trying to modify that with synthetic peptide proteins, okay? So what do you do to ensure that the bioactive compound is delivered at the, okay, I think I did mention that for targeting. We have not tried targeting strategies in our lab, but that is something which we will now work on. Can nanogel be an app delivery system to target a drug having low oral bioavailability? Probably. What is the next question is, 1 second. Please tell the possibility of a self-propelling nanoparticle. Yeah, that's a very interesting question. And in fact, my colleague at IC Bangalore, he has done a lot of work with this, and we are trying to do something with this also. We can have enzyme activated self-propelling nanoparticles which can release CO2 and which can you should read up work from Amrish Ghosh's lab, from IC Bangalore. He has done a lot of work on that, and there are a lot of other groups in the Europe and US also which has done that. Also, please let me know factors that has to be considered for optimized for efficient formulation. I hope in the talk I have addressed all of this. So this looks like questions before I was at that part. Liposomes used in TB, tuberculosis, and how are they administered? To be honest with you, I will not know the clinical way of administering. I think by injections. For TB, they might have some oral capsules maybe, and also maybe by by like nasal something. I'm not sure. I'm really not sure. Is chitosan an efficient nanocarrier? Yes, because it has a positive charge, and as you know that siRNA is negatively charged, so it is used for complexing. So it can be used, chitosan can be used. Which one is high toxicity nanoparticle, nanorods or nanowires? Interesting. It would depend on the dimension of your nanowire and the nanorods. Because nanowires will have a very thin, because it's a wire, very thin height, it might be more toxic. But of course, it also depends on what is the length, right?
Now?
Dr. Neetu, anyways, yeah.
Yeah, I think our turn.
Thank you so much, ma'am. Now we'll be departing for lunch break. An afternoon session will begin by 2 p.m. IST. So we hopefully will meet you by 1:55 at least. Thank you so much.
Thank you, madam. Thank you so much.
Thank you. Okay, bye-bye. Bye.
Dr. Sudha? Dr. Sudha? Hello. Yeah. I think we can meet around the 1:55. Okay.
1:55. Okay.
2 o'clock we can start, no?
Yeah, yeah.
Okay.
Yeah.
Okay. Okay. See you. Bye. Dr. Sudha,
Yes.
can we start?
Yeah, please.
Okay, thank you. Okay, start.
Hello everyone, once again welcome you all. Hope you had a great morning session. Now let's resume to our next section. So moving on to the next lecture, we would like to invite Dr. Prakash BK, Assistant Professor, Department of Public Health and Community Medicine. His primary areas of research interest include epidemiology, community health workers and health economics. Over to you, Dr. Prakash.
Thank you very much. A very good afternoon, everyone. And I'm honored to invite our distinguished speaker of this afternoon, Dr. Devasmita Alon. Dr. Devasmita Alon is a renowned academician and researcher from School of Biological Sciences, National Institute of Science, Education and Research, Odisha. She is currently heading the Molecular Genetics and Epigenetics Group at NISER, Odisha. She has received her Master's in Biochemistry from Nagpur University and earned a PhD from Banaras Hindu University. Her graduate work focused on molecular genetic analysis of RAB11, a small GTPase, along with characterization of L3TB, a novel tumor suppressor gene in Drosophila. For postdoctoral work, she joined Professor Howard A. Nash at Laboratory of Molecular Biology, National Institute of Health, Bethesda, USA. Her work during her postdoctoral research was aimed at unraveling the molecular targets of general anesthetics using copy number variation. After joining NISER in 2010, Dr. Devasmita focused her research towards understanding molecular genetics and epigenetics of various aging and neurodegenerative eye disorders like glaucoma and cancer. In the time when we are talking a lot about aging associated diseases and neurodegenerative diseases, her research is of a very prime importance. She has published several publications in renowned national and international reputed peer-reviewed journals. Today, she will be presenting on the topic natural products as regulators of tumor suppressor genes. On behalf of organizing committee, I welcome Dr. Devasmita Alon for the presentation. Dr. Devasmita Alon, now the podium is yours. Thank you.
Thank you, Dr. Prakash. At the outset, I would like to thank Professor Mohanan also for the kind invitation. So I'll just present the slides and see whether it is working, and then we'll go ahead. Is my voice audible to all?
Yeah, it's very audible.
Okay, so let's go ahead.
You share the slides, please.
Okay.
Okay. Yeah.
Okay.
Yeah.
Okay.
Very nice.
Okay.
Dr. Sudha, Dr. Sudha, it's visible there, right? Dr. Sudha? Dr. Sudha?
Hello, it is visible.
Yeah.
It is visible.
Okay, fine.
Visible and audible. Okay.
Dr. Devasmita, you can go on.
Okay. Thank you all. Thank you for the kind introduction, and let's start the webinar. Actually, when Professor Mohanan asked me about presentation, I thought a topic of natural product will be relevant to the theme of the conference. And as you can see in the, I'll just start the pointer. So as you can see as you can see this place, it is actually nicer. It is a residential campus around 35 kilometers away from the city of Bhubaneswar. And we house almost like 700 to 800 students who come here after their +2 to do their integrated masters. Then they do, some of them have integrated PhD and the PhD program. They are enrolled into it. And School of Biological Sciences is this building where I work. I would have normally talked from there and but today I am not there. So I just wanted to show this place so that, and you all are welcome over here when the situation gets better. And as Dr. Prakash said that I work on different other projects in the lab, but today I'll be focusing on the natural products and how they can actually be used as a regulators of tumor suppressor genes. So what are natural products? So natural products are basically the ingredients that we eat with the food, right? Any living organism have these natural products, and our food always have these natural products which gets ingested when we take our normal food. And the tumor suppressor genes are as the name indicates, suppresses tumor in the body. Normal gene function, if they in the normal body, these genes are always regulating and they keep the tumor in the suppressed condition. Okay, so with these two keywords, we will move on to the next slide. I hope I'm doing it. Sorry, but it just doesn't go to the next one. Okay, so I just have to move to on the right side. Okay, then I got it. So and this is the introduction slide where you can see the product curcumin, which is seen in the turmeric, right? On the spices that we take, it has curcumin. And with the green tea, this is EGCG, and the soybeans have genistein. These are all various natural products which are known to actually inhibit the oncogenes and activate the tumor suppressor miRNAs. So what will it do is that, as I said, in the normal body, a tumor suppressor gene would normally keep the tumor in a suppressed condition. So loss of function of this tumor suppressor gene can lead to tumor, and if we do not have this tumor suppressor gene working, so you can have cancer in the body. So this much is enough for the undergraduates. I gather that there are a lot of undergraduate students also in this audience, so I wanted to give this brief overview to just follow the talk through. Okay, so and the natural products that we are talking, just a small list of it. If you look into the literature, you have thousands of different products that has been isolated from the food products, which are actually work as various anti-cancer agents. Here we are talking about the cranberry, cranberry, the algal alkaloid, artemisinin, and ashwagandha. Okay, so then I'm sorry for this, but I don't know how to go to the next slide. I'm using Zoom for the first time for the presentation mode. So if somebody, oh, I got it. Okay, thank you. So the next slide, I'm in the, so we just are in the introduction part where we are talking about how natural products can be acting as regulators of tumor suppressor genes. So the focus of the lab when I started in 2010 was basically looking at glioma. What is glioma? It is a brain tumor which is most aggressive and lethal. The overall incidence of glioma in USA is about 6 cases per 100,000 individuals, with glioblastoma accounting for 50% of the cases. So median overall survival is 15 to 23 months, and the 5-year survival is less than 6%, which is the lowest long-term survival rate of malignant brain tumors. So next slide. Is actually what is the cure for that? Till today, there is no cure for gliomas. As I said, the median life expectancy is within 5 years. So it is really disturbing for the family members. And the cure is basically what we know is that there is a surgical resection, so that there is, you remove the tumor, then there is radiotherapy and chemotherapy. But if a single cell escapes this chemotherapy, it can lead back to tumor. So those are the condition where we stand today. So who comes to our rescue? So basically we are talking about plants, and these plants, can they actually give the natural products which can allow us to actually cure these tumors? So as I said, when I started my lab in 2010, I didn't had a PhD student. We started all the work with a master student, Saurajit, whose work I won't be presenting, but that is the background of all the work that I will be showing today. So we started with artemisinin and curcumin, and one artemisinin is nothing but the Artemisia annua plant product, which is used in the traditional Chinese medicine. It is a primary candidate for the drug repurposing and is an anti-malarial drug, as you may know it to be. So it induces cytotoxicity in liver, gastric cancer, colorectal, lung, breast, cervical, ovarian, and esophageal cancer. So, and to look into all these things, we cannot use humans, right? Then we need a model organism where you can model this tumor and look for the effect. So I started with my PhD experience with fruit flies. We started to look at fruit flies. What are fruit flies? All the name indicates Drosophila. They are actually you can see it in the rotten bananas. If you have some bananas, if you keep it for more time in the house, you can see some flies hovering around it. These are the fruit flies. They are holometabolous insect. Holo means whole, and metabolous means entire metamorphosis is completed. So it has 4 life cycle: embryo, the larval stage, the pupal stage, and the adult. So what happens, what is good part of it is you can see the third instar larvae, they are actually transparent. You can look it under the microscope, and some structures in the larval larvae are kept aside to make the adult structure. These are called as the imaginal disc, and these are actually, you can actually create a tumor in the larval stage and follow it till the adult stage. So that was the advantage of the fly model and the expertise that I had. So we started screening for these natural products in by creating a glio by using I should not use the word creating, because we used a fly model which was available, and I'll come in a bit to that. Okay, so is that clear till now? So we will go ahead from here. Okay, so now I'll come to dihydro. So I think I missed one slide in between. So to understand the effect of the drug on the tumor regression, we used a fly model L2GL. As you can see, the L2GL stands for lethal 2 giant larvae gene. 2 comes from the second chromosome. All the Drosophila genes have been named based on the mutations. What the mutations phenotype it will give, that has been named after like that. If you have a lethal, the mutation is in second chromosome, you get a giant larvae, and this larvae, it dies at the larval stage. So this is the loss of function of this tumor suppressor gene led to a brain tumor. And in the work which we published in 2014, we can show that both artemisinin and curcumin could inhibit tumor growth. So what is this L2GL? What it does in the human? See, it encodes for a protein P127, which is required for the apical basal polarity, and the human homologue is LGL1, and it is decreased function of LGL1 causes colorectal cancer in human. So homozygous mutation leads to larval lethality, which is cured by the artemisinin and curcumin. Okay, so this is this slide is busy, but don't worry, I'll just explain it out. But from here, you just have to understand that there is a human homologue of this gene. So it is not that we are looking for a gene which is not present in human. People do ask, if you do a work in the fly, how is it translated in human? Right, so that is the question that we are talking about, but it does create tumor in the case of human also. That is the gist that I want to present here. And as I said, there is a life cycle of the fly. What the work that we did in 2014 falls short of is that though the tumor was rescued, the flies never emerged at adult flies. I'll come to that, because if you look at Drosophila L2GL mutant flies, they are basically homozygous recessive. They are basically recessive genes, right? So when you have 2 copies, they become homozygous, and you should see a tumor phenotype. These are lethal. And if you are in the heterodygous condition in which the flies are grown, I'll come to this again with, if somebody doesn't understand with this much background, I'll come to a background slide also at the later stage when how we are setting up the cross. Okay, so there what happens is that the adult flies never got rescued with artemisinin and curcumin. So since this study had never been, and the fly model was not used till then to understand about brain tumor, there were other flies have been used for many other model organisms as a Maria. So we published this work in 2014 to just give a sense that, okay, there is some rescue, but we did not understand at that time why it did not give us a real homozygous fly, which could be adult fly, could be there. So what did we do at that time? I'll just go to the next slide. We went to the next metabolite, means the metabolite of artemisinin, dihydroartemisinin. The reason was that because we knew that from literature that artemisinin had poor bioavailability and a very short half-life in vivo. So we thought that maybe by the time we feed the flies and they were not able to get the enough product to rescue the effect. And the other theory that has been discussed in the paper is that this mutation also affects ecdysone receptors. So if ecdysone receptors are affected, so maybe they were not able to metamorphosize properly. So with this thing, two questions, we left that paper at that stage, but we moved on to another product, which is dihydroartemisinin, which is a semi-synthetic derivative of artemisinin and has anti-malarial activity. And like artemisinin, anti-cancer activity has been studied and shown against pancreatic, leukemic, osteosarcoma, and lung cancer in dihydroartemisinin. But in all these studies, if you look into, the brain tumor were missing. So we asked the question, can we use DHA to escape pupil lethality that we could not do earlier? And can it cure the brain tumor in case of L2GL homozygotes? So then this is what I wanted to say, that this is the culture, the fly culture. Those who have not seen flies, they are very good organisms to study. You can see this is a male and this is a female. Characteristic pattern, pigment, segmentation pattern and blackness because of the fusion can be seen in males, which is not there in the females. So when you cross these two flies, you basically get three different products, right? One is that of L2GL by Curlio, another L2GL by Curlio. So 50% of them will be L2GL by Curlio. 25% will be homozygotes which lie as larvae, and another 25% die as embryonic lethal. So with that, actually, we went, and for those who want to take up this research or anything, there is a marker in the larval stages which can distinguish the heterozygous and the homozygous. That is, the Malfi gene tubules of the homozygous larvae are actually white in color, but the heterozygous are yellow in color. So you can distinguish between the heterozygous and homozygous in the larval stage. So the experiments were planned in 4 different sections. One was morphological, where we could look at the brain tumor and say whether there was a regression in tumor. Then there are anatomical, then health span and behavior. I'll be going each of them one by one. So looking at the morphological effect of this drug, since this DHA had not been used for brain tumor, we had to find the minimum effective concentration in which the drug was effective. On the right-hand panel, you can basically see, I'll just go to the wild-type larvae first. This is a larval brain, and these are 2 optic lobes, and these are the ventral ganglia. If you compare between the wild type and that of the L2GL flies, you can see there is an enlargement of the brain, and this is what we are calling as a tumor in the flies. And when you treat it with dihydroartemisinin, you can see that the size has actually reduced back to that of the normal. So this was our essay, to first look at it. If we can get the adult flies was later on, but we dissected the larvae at the 3rd instar stage, and we used to check whether we are able to rescue the brain tumor. While looking at the tumor, it is very important that there should be no toxicity effect. And Drosophila again has proved to a very good model to look at the in vivo condition. So in the lower panel, if you look, DHA, when treated when the wild type flies were treated with DHA, they should not have any adverse effect if you can use it as a drug in future. So that experiment was also done, and we could see that the effect was only restricted to the tumor cells. Here I cannot say tumor cells, but I can say that at least it is restricting the proliferation of the this region so that it is not so big enough, right? So morphology can only tell me that much. So if you plot this graph of the concentration of DHA use, you can look at it. We calculated this brain area, which is in 10 to the power 3 square microns, and the drug concentration in micromolar, you can find that around 250 micromolar, we got a minimum effective concentration of DHA, which we have used in the subsequent experiment. Here is a plot of an untreated larvae, untreated L2GL brain, where you can see that the size is around 150. And here it will be almost like 100. Okay, so that is 180, I should say, not 150, more than 150. So it is 180. So this was a difference we were calculating and assaying at this point, right? So 98 is what we are talking about. And when you rescue it out, it came to around 100. So, and we cannot do this experiment in just one larvae and check it out, right? The N number is very important. As students, you all should also know that this is very important, that a critical number, statistical significance has to be obtained. So we talked around 30 larvae. And the inference from this experiment was that dihydroartemisinin effectively inhibits tumor growth in L2GL homozygous larvae at 250 micromolar concentration. So this is what has been used in the subsequent experiments. So now, just because I told that the tumor size has actually regressed, does it mean that inside which cells is it killing? We did not have any idea about it till we did the BRDU assay. BRDU is an analog of thymidine, and as you can see, this is the optic proliferation center. This center is very characteristic for the replicating zone in this brain. You can mark that with that of the BRDU and look it under the fluorescence microscope. If you take the wild type, you see the very nice proliferating zone over here, optic proliferating cells in a zone, but you do not see it in the L2GL. It has all migrated in different regions. Now, when you talk about DHA, it is actually able to rescue that effect back. So this was very interesting, and we thought that actually DHA is targeting to the required, DHA is targeting to the required cell, which should be tumor specific. And so BRDU staining assay of the third instar L2GL by L2GL larvae with brain tumor reared on dry hydroartemisinin shows revival of concentric rings of replicating cells. So we were able to rescue it both anatomically as well as morphologically. Now then when one would like to know what is the mode of action, right? So what we did was that there is a lot of studies in literature that in a normal cell, you have, let us say, 100 moles of reactive oxygen species. When you take a tumor cell, this amount is increased manyfold. And the whole point is, can we actually check this reactive oxygen species and see what is the condition? And it has been seen that if a reactive oxygen species goes beyond a threshold, in more than the tumor cell, it will actually become apoptotic and die. And that was our essay when we took dihydroethidium and stained this one to look at the Ross species. And I will not go through the detail of all the plots here, but if you compare between the wild type and that of the tumor cell, this is the tumor ones, L2GL by L2GL. On treatment of GHA, you could see an increase in the concentration of in the amount of ROS. So we think that the mode of action of dihydroartemisinin is ROS mediated. Now coming to the major thing that, okay, it was, we could rescue we could actually show anatomically and morphologically that was a rescue, and we could know the mode of action. So what happens to the flies? Are they able to survive the same amount of time as that of the wild type, or are they able to only rescue to certain point and then they die? So we are talking about drugs, right, which will be making the fly healthy and not making them more sick. Even if the tumor is actually, you say the tumor is regressed, but the fly became sick, then there is no use of that drug even. So in that case, what happened is that yellow line, if you look into it, these are the L2GL larval plot. If you look in the percentage survival, and when you talk about artemisinin and curcumin, we have both done in the 2014 paper, whoever is interested can go and look into it. We have done the synergistic effect also, but because it is too crowded, I didn't plot it here. You can see that these artemisinin and curcumin actually enhance this longevity little further. But when you talk about DHA, we could see it was almost like that of the wild type flies. And this is what we are talking about. The L2GL by Leo actually in the heterozygous condition have curled wings. So you can look at the wings and say whether they are heterozygous or homozygous flies. And here in case of, this was the first time, I think it was in 2016, not 15, it was 2016 when we got these flies, and we were so excited in the lab because 2010 when we started the work, we wanted to get a fly which could be rescued with that of the drug. And this is the story stand today, that we were able to rescue that with the dihydroartemisinin. So homozygous L2GL deficient larvae reared on DHA rescues larval lethality, ultimately leading to the emergence of adult flies. Okay, so that is okay. So now the question comes is that what is the behavior? So now if the flies are healthy and they have actually come out, are they really healthy? What is the aspect of their health? Are they able to walk properly? Let me put this effect for students who are there. An observation in research is a very important aspect of research. You know, these flies of L2GLs actually have been in the field for more than 100 years. But the sluggish movement of locomotion which is observed by the L2GL flies had never been reported till date when we published it in 2014. And you can see that these are basically agar plates which have been food has been, coloring agent has been put, and the larvae are actually third instar larvae are allowed to crawl. And when they crawl, we calculate the distance covered in 30 seconds, which is in millimeter. And if you look at the x-axis and the y-axis, the wild type, these are able to go around 25 millimeter. But in the case of L2GL, we could find that they could only go till that of the 12 or 13 millimeter. This is an average of that, but you can see there is a range. But when you treat it with DHA, it was almost comparable to that of the DHA, comparable to that of the wild type. So this actually helped us and boosted our motivation to further go ahead. But with this experiment, we actually could say that the sluggish locomotory effect of L2GL homozygotes were rescued with that of the drug. Then we went on and looked at the adult flies because this was the first time we could actually rescue the effect. So the flies were also very critical. So we wanted to check, this is basically two vials, Drosophila vials having the food. And you can see they were allowing the fly to go beyond a mark, one at a time. And you can see that when these are plotted with the wild type and then the L2GL, drug-treated L2GL, there was no difference at all. Both of them where we did not had a heterozygous, we did not had a comparable L2GL by L2GL homozygotes to actually plot here because there was no fly till it could be rescued. So the experiment is such that we actually see that there is no difference between that of the wild type and the DHA treated homozygotes. So the inference that was drawn was basically newly emerged L2GL homozygous flies have survived larval lethality and persisted with normal locomotory behavior. Actually, so I'll just go to the next slide. I actually see 2-3 questions coming up. Should I answer right now or should we take it at the end?
You can take it at the end.
At the end, Prasamona. Okay, thank you. So, so what happens is that if you look at the tumor, if this drug is effective, if you take out the larvae out of the drug, the tumor should reoccur. Okay, and that is actually what we saw, that with the sixth transfer when we did with and without drug, we could see that the tumor was recurred back in the flies which were not reared on DHA. I think there is a typo here, sorry. Tumor recurrence in progeny of L2GL deficient homozygous fly not reared in DHA. So that these flies there was a recurrence of tumor. You can see blisters in the wing, and if you open it up, you can see tumor has come back. So this was the part done through the screening of the screening with in the case of flies. I'm putting the success story here, but along with artemisinin, there were a number of drugs that were tested, and many of them did not work. Why I should say many, none of them worked except for artemisinin. So I'm putting for students who are there in the audience, the faculty goes and talks about the success stories always, right? But then on the background in the lab, there is a lot of work that goes on, and one should not be discouraged, because from there only you get a positive effect or a positive research going on. So then we went on, because the fly model gave us a positive hit. So we went on to look at these DSA in the rat brain tumor model. For the rat model, what we did was that we took the C6 cell line, which is a glioma cell line, and injected in the tumor induction in the SD rats. And so what happened, we took 3 million cells and put it into it. It was a subcutaneous injection in the right flank. And when this was administered around 15 to 45 mg per kg, and we were looking for the regression. And the study is still incomplete. We are looking for the gene expression study, which we have not done. So I just gave you the plan of work which we are following. So what was the story behind it? So when this tumor was grown, the C6 cells gave us a tumor phenotype. And in that tumor, if you look at it, when you have actually 0 mg per kg per day body drug was given, according to body weight actually of the mice or the rat, you give the drug, right? So then you can see that there is a tumor. At 15, you can see a regression. At 13, you can still see a regression. Around 45 or so, it was almost that of the lower volume. So treatment that was started from the first day when the tumor volume was around 100 millimeter until 14 days. So you can see the tumor has regressed. So what we came up with that treatment with DHA inhibits tumor growth in the rat glioma model. So this is because we were all researchers would like to, when we are doing the experiment over the days, we were using vernier caliper to actually measure the tumor. So after the days are over, you sacrifice the animal and you can look at the PBS that is displaced by the tumor, and you can look at the tumor size also. And here you can see there is the size of the tumor. At 15, it is reduced. At 30 mg per kg per body weight per day also, there is a reduction. But the best part that came out of it is that we did not see any identified tumor when we used 45 mg per kg per day in the experimental group. So this shows that we can translate a drug which we can screen in the Drosophila model or any other model. People use today's iPSCs also. People use different kinds of model. Drosophila is not the only model. You can screen it and then bring to the rat model and look it out at the mice model and see. So with this, the inference that we came up with that DHA rescues subcutaneously bearing glial tumor at a concentration of 45 mg per kg per day. Then it is equally important that we should know that just like the flies, the rats are healthy. So we looked at the body weight of these rats over the days when this treatment was being done, and you can do not see a drastic change. If there was a drastic change, the experiment had to be aborted in between. Okay. So, and the urination, defecation, the water consumption, the food consumption, and crawling behavior all were recorded, and there was no drastic change on those. So we said that with that we inferred that DHA had no side effect even at the 45 mg per kg per day body weight. Then tumor was very small when we took this 100 mm cube, right? So we increased the tumor size, let it grow for some more days. And when we started the medication, we saw that not only at a 100 millimeter cube, you can rescue it till it was 500 mm cubes also. So that was very promising, because you will not get everybody at the small brain tumor size, right? Somebody will have a smaller tumor, somebody will have a bigger tumor. So in that case, whether this drug will be effective or not, that can be checked out. So there was no identified tumor observed at 45 mg per kg per day in the experimental group. And the inference that we draw is that DHA rescues subcutaneously bearing glial tumor at a dosage of 45 mg per kg per day, even if the tumor volume is large. For us, we thought that for 500 mmQ was a large size. But the question is that one can keep on doing at a much larger size. 250 also worked for us, but my student was not in in a mood to go for all the 5. We said that can we do it at 750 and 1000? They said, ma'am, that is enough. We should go ahead with the mechanism part now. I said, okay, that's fine. Let's go ahead and look into it. So we looked at the angiogenic marker. For any tumor to progress, you need blood to be poured in, right? So that is a process of angiogenesis. New blood vessels are formed, and CD31 is a marker for angiogenesis. And we looked into the CD31 expression in all of them. And I'll just go to the next slide where we have quantitated it. The microvessel density, if you quantitate it with the CD1 positive cells, you can see that from that of the 0 where there is no drug to that of the 45 mg per kg per day, the microvessel density has actually decreased, which also indicates that the tumor is regressing. So DHA inhibits in vivo angiogenesis, but this was already known for DHA. It is not that we are saying that DHA in vivo angiogenesis was, but there are some literature which were done in the cell culture model where they had shown this effect. We are showing it in the in vivo model. Now, the other thing comes is that when you do an experiment, when we are doing it subcutaneously, we are not doing it at the right place or location where the tumor was supposed to be. So we did some orthotopic injection where the brain tumor was, cells were injected in the brain, and we have been able to create the tumors in the case of the orthotopic also. There is a cell number, standardization has been done. The days that are required for the tumor growth has been known, and the days for the drug treatment is also done. So if you look into the orthotopic experiment, I am not saying it is at a full, see most of the work which I'm publishing today is not published at all. So, but this is the one that is the standardization part where we are showing that if you look at the tumor cells, when the number you talk about, you can see that in case of 8 to the power 10 to the 5 cells, it is effective to go for higher to look at the effect. And when you take this with and without DHA, you can already see an effect against that of the without DHA and that of the DHA. Okay, so the study conclusion till now is that the DHA gives complete rescue, thus generation of adult flies at 250 micromolar concentration in case of flies, and there is a rescue of subcutaneously bearing glial tumor with DHA at a dosage of 45 mg per kg per day. So these studies are ongoing, and we are repeating the orthotopic implantation of the glioma cells following drug treatment to check for the survival rate. And also the ROS measurement, which we said that, you remember I said that it is the mode of it which DHA work. So if it has worked in the fly model, we want to see whether when we do the ROS measurement in case of DHA treated rats, can we understand, is it actually ROS mediated, and can we go to the mechanism? Not only is that, as I showed in the protocol slide, flowchart of that, we want to look at all the gene regulatory pathways and the expression of the genes to say which pathway does dihydroartemisinin follow. So but that is another very big question in the field, right? In the anti-malarial field also, people have been using artemisinin and dihydroartemisinin for a long time, but there are a lot of debates about what is the mechanism in which dihydroartemisinin works. It can be very different in case of tumor than it is in the malarial drug effect, but we would like to understand what happens in this case. So no work is possible without students. Means today I am sitting over here and discussing about here because somebody in the lab has actually toiled hard to get this to the place where I can talk about. So this is my lab with my research students, PhD student. Unfortunately, neither Saurajit or Bhavishya, who did this part till now are here. They have all graduated, and they have moved on from the lab and joined different positions for research positions. But Shushri is my PhD student who is actually taking up this work, and she will be following on looking at the mechanism. And I have been generously got grants without money. In their case, no research, so has got grants from various agencies, and I really acknowledge them. And thank you for a patient hearing and giving me the time, and this floor is open for questions. I think I'm in time, right?
Madam, you can able to read the questions, or
I will read it. I'll just go, Professor Mohanan, I have not used Zoom till now, but I will try it out.
Okay, Otherwise we can read it from here, you can answer it.
I'll just try it first. If it doesn't work, I'll just
Sure.
I take a class on online Zoom,
Okay.
but that is something which we don't present anyway. I just.
That's okay. You try. Otherwise,
I got it. So
okay. Fine.
Thank you, Professor. So first question is Archana Kumari. She has this question: is that why Drosophila used as test system as brain morphology is quite different from humans? That's a very good question. I'll come to that. The second question is, will reproductive study also be relevant in reproductive test toxicity study? Can it be replacement for mice and rat to any extent? Okay, those are very relevant questions. So thank you for the question, Archana. As at the outset I said, if you look into the Drosophila, the life cycle is very small. Within 14 days, you are actually able to grow it from that of the from that of the embryo, the egg, to that of the larval stage, and within say another 14 days you have an adult fly. You do not have those kind of liberty with that of the rat, and this all needs money, right? If you screen something in the rat model directly, can you imagine how many rats we have to, or rat or mice we have to sacrifice? We do not have that kind of liberty. And as I said in the beginning, I was starting up my lab, right? I did not had any resources. I had got only, I should remember that, I had got 5 lakhs to run the entire lab for 2 years. Okay, so you can imagine under those conditions when we started, you really need to think that what is doable. And that is how we started with it. Yes, the question is valid, whether what we are getting right now, is it that what we will be able to able to replicate it? Yes, we could have failed terribly if we could not have replicated in the rat model. And I'm not saying that any drug that we can get it validated in the fly model, that will be valid in the rat model also. But for if you have to get number of screens, that you want to screen a number of drugs, you can do it very easily with the flies. Within 5 to 6 months, you could have screened almost like 15 to 20 drugs. And later on, you can take it to the mice or the rat model to look at the effect. So that would be why I would say that Drosophila used in the stress system as brain morphology quite different from humans. That is true. And that is what we are aiming at when we are looking. So I did not show you the histology slides which were using an hemocyanin stain for that of the rats. That is our aim, that whether we are able to, we will be able to replicate it in the rat model. Now the question is, will reproductive studies also be relevant in reproductive toxicity study? Yes, Drosophila has been used for number of toxicity studies. I would not know the details of all the studies that have been done. I would not have followed it up, but you should look into the literature and see it out, whether it is specifically done to the question that you are trying to address. Okay. The third question is, can it be replacement for mice and rat to some extent? I would say certainly. If it is a screening of the drug, yes, it can be done. But later on, there is false positives that can come in the fly screen that you can have to avoid in the later time. Okay, so which you can be actually, if you take it to the rat model, if you get an effect, it is fine. If you do not, you have to stop it at that time. But yes, it can be used for the screening purpose. Okay. I will just, is there another question? I see 4 questions and I have 3 here. So how do I get to that? I'll just check it out.
Question is the last one.
The last one?
Yeah, last one.
It is all is done?
Yeah, no. At which stage tumor the larvae was exposed to drug?
The drugs were effect. I could not see that. I'll learn it later on, Professor Mohan. But the question is that they were exposed right from the beginning. They were exposed at the embryonic stage itself, and they were allowed to grow on the drug itself. Since there was no side effect of that, so it didn't matter to us. But yes, there can be some time specific things that can happen, and you may not be able to see it directly, and you may have to, because it was a larval tumor, we could do it at that stage. For example, let us say if the tumor was in the adult stage, but you have to look at different time zones. We have done those experiments. For the time's sake, I think it is there in the 2014 paper. We have done it at the larval stage and looked at the effect. The more you come near the stage, the tumor has grown at the larval stage, the more the bigger the rescue is less. So it depending on that, we could see it at different stages also. Any other question?
No more questions, right? Yeah.
Thank you so much, Dr. Devasmita, for your valuable time, which was very informative and interesting. So, from your lecture, we were able to understand the possibilities and drawbacks, which of course hasten the development of effective and safer cancer preventive and therapeutic drugs for clinical use. Thank you once again for your valuable time with us.
Thank you so much. Thank you. Thank you so much. I don't know your name, but thanks a lot for your kind. Thanks for the comparing.
Thank you so much, Dr. Devasmita.
Thank you, Professor Mohan. Thank you, Dr. Sudha, Professor Sudha also. Thanks a lot. Thank you for the invitation, and we'll be in touch. Okay?
Okay, thank you.
Okay, thanks a lot. I can see the questions now. I learned it. Thank you.
Dr. Narisa?
Yeah.
Hello.
Yeah, okay, you're online. Okay, please wait 5
I am
minutes.
here.
5 minutes, please. Okay?
Yep.
How are you?
All good, thanks. And you?
How is the winter there now?
Weather is horrible.
Okay, just wait for 5 minutes. Okay?
No worries, I'm here. Yep.
Yeah, 9:30 of your time.
3 o'clock.
Okay, I think we can start then. Okay, Dr. Sudha?
Yeah.
Can we start? Can we start?
Yeah. Yes.
Okay. Over to Adira.
Welcome back. Now let's move on to the next section. Okay, now I would like to invite Ms. Harati, who is going to compare the. Thank you so much.
Welcome back, everyone. It's time for our last lecture of the day. We invite Dr. Sachin J. Shenoy, Scientist F, Division of In Vivo Models and Testing, Sri Chitra Tirunal Institute for Medical Science and Technology, to introduce and invite our next speaker. Dr. Sachin's area of specialization includes experimental animal surgery, anesthesia, and perioperative acute care. Over to you, Dr. Sachin.
Thank you, Arti. It is my privilege to introduce Dr. Annalisa Torella, lecturer in Division of Pharmacy and Optometry, Faculty of Biology, Medicine and Health Sciences at University of Manchester, United Kingdom. She received her PhD in Materials for Environment and Energy from University of Roma, second, developing a 3D bioprinting system. Dr. Torella's principal research interests are in the field of polymeric biomaterials and their processing via additive manufacturing technologies. The focus is on combining functionalized and bioactive materials like hydrogels, including nanoparticles and cells, with biofabrication technologies to develop tissue engineered and therapeutic technologies. The other research activities of Dr. Torella include design and manufacturing of nanotechnologies for the controlled and or sustained release of drugs and nucleic acids. Dr. Torella's research group works at interface with multiple disciplines in two main research areas. First, in manufacturing of nano and micro technologies for drug delivery, and second, in design of bioengineered in vitro 3D models. She has several outstanding publications in national and internationally reputed journals. Now she'll be talking on the topic hyaluronic acid based nanotechnologies, targeted delivery in nucleic acids for CD44 positive cancer cells. So on behalf of the organizing committee, I welcome Dr. Annalisa Torella for the presentation. Over to Dr. Annalisa.
Thank you. Thanks for the very nice introduction. And it's a pleasure to be here and being invited to discuss about what we've discussed and discovered in our research group over the past years. So I will just move, conscious of time, to my presentation. So let's see if it does work. I hope you can see my screen.
Yeah, we can see the screen.
Brilliant. So let me just move slightly the video. Okay, so today I'm going to talk, as introduced, to an experience we've learned over the years. So it's a research that lasted about 4 years, big project on the design of nanotechnologies to get a target delivered to CD44 expressive cancer cells. So before getting there, I've got a really nice introduction, but I give you a bit of my background. So I've studied and I did all my studies in Pisa as a biomedical engineer. And then, yes, I moved to Rome where I could combine what I learned as a biomedical engineer to more active principles in the design of a manufacturing device. And at that time, I'm not too old, but it was already 10 years ago when I defended my PhD, and 3D printing systems were at the very beginning of the early stage. And in particular, there were not too many commercially available tools at that time. And we were between the very first discussing the arena about printing cells and including them in gels and see how it was possible to facilitate the regeneration of tissues. Then I took a challenge, then I moved back to Manchester where I did part of my PhD project. And I wanted to focus more on nanomedicines and nanoparticles because I thought that together with 3D printing, it was another really big area of interest and probably with many future applications. as you may know. And I had the chance to really build up my research group. This is a part of it. I think most of them are already gone and graduated, but that's when everything started. So my research group now focuses on too many different research areas. Today, I'm going to talk a bit about nanotechnologies and how we design and manufacture them. But also we learn the experience and we apply the use with the other part of the group that it's more interested to use biomaterials to engineer them to replicate the 3D in vitro model. And in doing that, what that is the main goal of the group is to kind of reduce the time that we have and we're facing now between the design of a new drug to its application to the clinical stage. From experience, we all learned over the past months, we know that the time has incredibly squeezed between the design of a vaccine for COVID-19 and its implementation to the clinic. But that was just because many products were already approved and they were just repurposed, so you could really speed up that phase. Otherwise, the gap is really large. So when we want to design something that wants to target and be used to treat tumor, that's quite very challenging. And that's because the tumor microenvironment is very complex. For me, it's even more complex because I'm a bioengineer. So I see the biology as something that is really puzzling me. It's a black box and I don't know where to go for. So I had to learn a bit more about it. Just to keep things simple and short for the purpose of this talk, when we try to design nanoparticles, what we learn is like they have to be small enough to reach the tumor site because mainly they're injected intravenously. But when they get to the tumor site, they have to be still active and be stable. So we need to find a way to protect the drug that we want to deliver to the tumor. Then so they have to be small enough to cross all the barriers and get to the tumor, but then being not too small to leave the tumor site. So we know already that there is a size range that has been identified between 50 to 200 nanometers. So that's the size of target for many nanoparticles. But there are very few successful nanoparticles examples that have made to the clinic. So the challenge is still there. So what we could do to improve their properties, the first thing that we need to do when designing them is to guarantee that it's a stable carrier, that protect the cargo against all the biological hazards that are present in the bloodstream, like enzymes, other oxidative factors, and so far and so on, hydrolyzation of bonds. Then we know that it has to promote the crossing the barrier of the tumor, potentially being internalized and retained within the cancer cells where they have to release their cargo. So you see that there are many steps that we need to design into nanomedicines. So what was our engineering approach? So the first thing, we look at the tumor microenvironment and we see what composes it. So we know that there are different cell types within it. Some are tumoral, some are healthy, some are slightly reprogrammed. Then we know that the composition could affect the permeation of such nanoparticles and also influence the behavior of cells. We know that we have nice models, animal models that could help us to predict it, but still they're not capable of reproducing it all. So we need to have effective in vitro models that could mimic really how to speed up to get to that clinic. Another thing that we need to consider when engineering it is the possibility that the tumor grows at a very late stage. So there will be an hypoxic core, another region with more oxygen. There is a blood flow, so the fluid of the blood that transfer the nanoparticles to the tumor, barriers to cross with different cells again. So what we can do, it's knowing which cells within this microenvironment have expressing some receptors that we could target. And we identified CD44 as one of the main receptors that is expressed, actually overexpressed in tumor cells, but it is also expressed across the whole body. So there are many other cells that do express CD44. Interestingly, and we will touch this point later, tumor cells do express more variants of these receptors, which change a bit in the length of a domain. So these variants are set are expressed on the membrane, and it is known that this receptor interacts well with hyaluronic acid. So very first thing that we would do as engineers is say, if I want to target that receptor, I need to have a nanosystem that is covered with hyaluronic acid. And in this way, we can hypothesize that there will be an interaction between the nanosystem and a cancer cell. Having said that, we need to identify really how this receptor can be an attractive molecular target for tumor therapy. As I said before, we need to hijack a bit the internalization of treatments, of drug therapies in normal cells, but just deliver that to the cancerous ones. There are different examples in the literature. What it is really hypothesized is that high molecular weight hyaluronic acid forms clusters of these receptors, which then pull in the larger molecule. And there are also other examples reported where exosomal parts of these particles in the bloodstream could be internalized as well, and that's how crosstalks happen between cells in the body. So building from that, what we said is like, okay, we could use this receptor and we could use hyaluronic acid as a vehicle for this internalization. The purpose of this project was about the internalization of nucleic acid. So one thing that we know was granted was that we want to protect this cargo, the therapeutic cargo. And the other thing that we took for granted was having hyaluronic acid on the surface. Now, the question was how we can bind this together and protect them. So we selected chitosanes as a candidate for doing so. We selected other positively charged polymers, but I just keep the talk very short. And we decided to include different variables on these chitosanes. That was why there was the selection. So we could control the molecular weight of the chitosanes as well as the degree of deacetylation, which means in a way that we could control how long was the chitosane and what was the surface charge on the polymer itself. In this way, what we hypothesized was we could create a complex of chitosane with a therapeutic cargo that would be stay together thanks to the electrostatic interactions. So nucleic acid are slightly negative, chitosanes are positive. So we could create a core and then cover it with hyaluronic acid. So this is what we did really. So we could have a kind of characterization of the chitosanes that we were going to use, and we start to combine them with both a small nucleic acid sequence, an siRNA sequence, and a slightly larger nucleic acid, an mRNA. And as you can see, we could successfully generate some complex with a size that was not too broad and quite monodispersed. We tested all this size and all this, the performances of the different chitosanes. And as you can see, the size is actually matching the requirements of nanomedicines themselves, as discussed earlier. They have to be injected in the bloodstream and reach the tumor size. What we notice is that with an increase of the nucleic acid cargo in size, so using an mRNA, particles were slightly larger, and that was quite easy to envisage at a very early stage. The other question that we wanted to check was if this complex was too strong to hold the nucleic acid, or if it was able to release the nucleic acid when internalized in cells. So what we did was an experiment that was looking at the displacement. So we were looking for adding another compound, heparin, that would take basically the portion of the mRNA or the siRNA instead of chitosane and then release. And we looked at how easy was this displacement. So how easy would have been for the cargo to be released when internalized in cells. And we saw that siRNA were really easily released, whereas mRNA would have a stronger interactions with the chitosanes, and just portion of it would have been released in the cytoplasm. So in essence, how this process works. So we believe that if we have a quite long and large complex of chitosane, we could add different domains of siRNA. and have a strong complex, but at least that we can control its release with a degree of acetylation. Then when we add the hyaluronic acid, and let's say if the movie starts, no, it doesn't, then we could cover this complex, so having still this core at the very center protected by a large and loose network of hyaluronic acid. Interestingly, it's like hyaluronic acid interacts a lot with water, so the size that we are measuring when it's wet is probably an overestimation of the actual physical size of the nanocomplex. Another thing that we wanted to check is now if we are at the stage to inject the nanosystems in the bloodstream, how would they behave when in contact with enzymes? So what we did was incubating the nanoparticles with an RNase enzyme, and then we check if they were actually capable of protecting the RNA or not. And we noticed that basically most of them were able to protect a portion of it, more than 80% of it, whereas the control, if my mouse comes, I cannot say it, sorry. Then the control of the sRNA, the scrambled one, was effectively degraded by the enzyme. So that was our control, that everything, all the system was working. We also tried to investigate if it was the hyaluronic acid or not that was protecting the cargo. And we did this quick test with the RNA and chitosan, and we noticed that it's not the hyaluronic acid that protects the nucleic acid, but it is the chitosan that, in a way, when it holds the nucleic acid, hinder the enzyme for its attack. At this point, another big question, it's the control of the stability of nanoparticles. There are many examples of nanoparticles that have to be prepared and immediately delivered because they're not stable. So stability was quite a thing to test. So what we did was checking the stability of size and surface charge of the nanoparticles. Storage was, so particles were stored for up to 2 weeks at 4 degrees. And we could notice that without the coating of hyaluronic acid, particles would be of a smaller size as predicted and would keep the same size. Without the hyaluronic acid, as you can see on the right-hand side of the slide, the surface charge, although, is positive. So again, if we would like to inject these particles in the bloodstream, we will have an interaction with all the proteins, and then we will have the so-called protein corona. So again, what we did was trying to check the possibility to coat it with hyaluronic acid. The size slightly changed as discussed earlier, but what we have is a stable coating that then gives a negatively surface charge to the particles. So we could hypothesize that there's no interaction between this nanoparticle and all the bloodstream and protein that are in the bloodstream. So it would reach the tumor site as it is. We also looked at the stability, as said, and we looked at how the ratio between chitosan and hyaluronic acid could protect it. And as you notice, we identified as the right balance between the weight ratio of chitosan and hyaluronic acid to be a 1:4 in weight ratio. So at that point, we could check that the size was slightly increased, but the zeta potential was very highly negative, and still particles were stable over time. Once we had a good control over our nanosystem, then we went to check if they were really capable of being internalized by cancer cells in vitro. So the very first test that we did was checking and comparing between chitosan nanoparticle and soluble hyaluronic acid. And the first test we did was the obvious off-target cell type within the tumor microenvironment, which are macrophages. So with the very first of interaction with macrophages, we optimized an in vitro model that could mimic a pro-tumor and an anti-tumor type of macrophages. So we have an M1 and M2, so-called in the literature macrophages, and an M0. And we looked at the interaction. So what did we do was just testing all the effective polarization of of macrophages, sorry, and be sure that we would have a robust model that was capable of expressing not only all the markers that are typical of macrophages.
Your slide is not visible. Can you reshare it?
Sorry.
Your slide is not visible.
Yeah, sorry. I don't know what happened.
You just reshare it. Yeah, it's coming. Okay, fine.
Yeah,
fine.
good.
Thank you.
Sorry for that. So let me take the keyboard. It's probably better than the mouse then. So having characterized all these models, what we wanted to look at was if they would actually express CD44, and they did. And we noticed that the M1 type, that is the pro-inflammatory macrophage that would be there in the tumor microenvironment, does express quite a significant amount of CD44. So not worth it, we also noticed that they would express the variant 6. And remember that we will go back to this point later on, but the variant 6, it's quite interesting in internalizing nanoparticles at the tumor site. We also looked at the internalization of hyaluronic acid labeled nanoparticles, and we could see that particles are bound to the membrane and then they get internalized. We did also a study where we could distinguish between the just internalized on the membrane and the nanoparticles that were internalized and releasing the cargo, and we could see that we could really measure that effectively in vitro. So the lesson learned was, okay, we do have enough target at the tumor microenvironment, so we need to check if it's possible to have an effective internalization with cancer cells. So at this point, we looked at cancer cells rather than macrophages, which were our target. We looked at just a big panel of cancer cells that they were all expressing CD44, and we did the first study with just soluble hyaluronic acid. And we could see that there was their internalization and effective internalization and localization with lysosomes. So we could prove that our hypothesis was correct. And we also looked at the internalization of nanoparticles. And as you can see, similar approach, we could also show that the release of the sRNA was proven with a scramble sRNA that tends to accumulate in the nuclei when it's released. What we also looked at was the profile of internalization of nanoparticles in cells. And so taking all the candidate of cells from the tumor microenvironment, we selected PANC1, HPC1, HCT116, and HCT29 because they were both expressing a lot of CD44 standard, but also a lot of CD44 variant. And again, we could see that we can distinguish a great uptake within the very first few hours when the nanoparticles reach the tumor. But then the internalization itself is a bit slower process, and it will take up to 24 hours to get there. And we could identify also that there's a sort of correlation between the internalization rate and the expression of CD44. So with higher expression of CD44, which leads to an increased internalization rate of nanoparticles, whereas a lower expression of CD44 leads to a lower internalization of nanoparticles. In essence, what we could see, if we have a tumor microenvironment with tumor cells and macrophages, then the tumor cells will internalize the nanoparticle much earlier than the off-target that are the macrophages. We also test how stromal cells, which are another important component in the tumor microenvironment, would interact with cancer cells. Again, we did compare firstly the expression of CD44 between these two cell lines, and we identified and selected a cell line that was expressing much more CD44 than fibroblasts. We looked at the lysosomal escape and internalization of nucleic of sRNA-loaded nanoparticles. In this case, is the sRNA that is fluorescently labeled, so we could track the cargo at this stage. And we did tests with both confocal microscopy and flow cytometry to quantify better the internalization of such nanoparticles. And we could see that after 24 hours of internalization, as you can see on the right-hand side, you feel a cloud of fluorescence across the cytoplasms, meaning that nanoparticles are slightly and slowly releasing the sRNA in the cytoplasm. So what we looked now was try to see the internalization rate of cancer cells cultured alone when incubated with nanoparticles, and of fibroblasts when incubated alone with nanoparticles. And as you can see in the plot, you can appreciate that the profile of internalization, looking just at the sRNA as our point of interest of measure, they have a very similar profile, which was kind of counterintuitive for us because fibroblasts would express less CD44 than the cancer cells that we selected for this study. And they also don't have a lot of CD44 variant 6 expression, that we couldn't detect it. So that was quite interesting. But then we decided to put these two cells in co-culture and see what was happening. So once we put a simple transwell experiment of cancer cells and fibroblasts cultured in proportion of how the number of cells you would have in the tumor, so more cancer cells than fibroblasts, of course. But if we look at the internalization per cell, we notice that now fibroblasts are not taking up nanoparticles. So there's a kind of crosstalk between these two cells, and when cultured together in exactly the same condition, then cancer cells just pick up and internalize more nanoparticle, even higher than what they could internalize when they were cultured on their own. And that was quite interesting and pushed up to start to do some in vivo studies. I never followed up them, sorry, so I cannot comment a lot about them. Another thing that we wanted to do was to check whether we could measure the efficacy of silencing of a gene of interest at that point. So the gene of interest that was selected was the KRAS. So we looked at the KRAS expression in the cell, so in SAT116, and look and compare with the treatment with lipofectamine on using the designed nanoparticle as carrier. So what we could notice is that the performances in silencing of the mRNA of KRAS expression was very comparable between the nanoparticles and the lipofectamine, which was quite nice. And also we noticed that was quite interesting and promising for the design of this nanosystem is that when we compare the performances of nanoparticles as prepared or after 2 weeks storage at 4 degrees, they keep and maintain their efficacy in vitro. So they have a very similar level of silencing of KRAS, of mRNA of KRAS. Another thing that we did was also testing what happened to the efficacy of these nanoparticles when they're exposed to an enzyme. In essence, when we have a selected sRNA sequence, what would happen to that? The one that we used before was a scrambled sRNA sequence. And in this case, we noticed that if we incubate with no enzyme, as you can see in the plot on the right-hand side, we keep exactly the same level of silencing, so really good efficiency. Whereas we have a slight reduction when the concentration of RNAs increases, but overall we keep a 50% silencing efficiency, which is quite promising for a nanosystem. So the very last experiment we did before moving to in vivo studies was to check what was the effect of perfusion of nanoparticles, because so far all the experiments were performed in static, but we know that our body is not static at all, but there's also some pressures and some fluids in any organ during our daily life. So we selected to include cancer cells, fibroblasts, and macrophages in a perfused system. What we did was check and assessing that cells would behave as we measured before, and so comparing static expression of CD44 versus dynamic expression of CD44. In this study, we included also another marker to check if there was a kind of reversing of the stemness of cancer cells, so we included the CD24 marker. And we set the flow rate to be very similar to what it happens in the tumor. So being very low flow, so keeping just the pressure, the interstitial pressure as reported in literature. So what we measure is like there's a slight change in expression between static and dynamic condition, as you can see on the plot. And this slight increase in dynamic condition is probably because shear stresses acting on cell membranes are kind of activating the receptor itself. So it's more overexpressed, but overall it's very similar. And we do see an expression of CD variant 6, as said before. So what we did again was trying to design this system to keep similar proportion between high number of cancer cells and a bit lower number of stromal cells and an even lower of macrophages. We incubated cells perfused with the same cell culture media circulating up to 24 hours. And in the media, we included nanoparticles with, again, labeling the nucleic acid, the sRNA. And what we noticed was that comparing to the static counterpart, the dynamic cells would increase the internalization. At this stage, we're measuring with flow cytometry, so we show that nanoparticles are in the cytoplasm. And we noticed that there was a really big increase of internalization. And at 8 hours, we could measure a peak of internalization rather than the 24 hours that we observed in static. We also looked under the microscope to be sure that we could see these nanoparticles. And as you can see, after very few hours, we could image and detect the internalization of such nanoparticles in the cell cytoplasm. Of cancer cells, fibroblasts do have their internalization, but we already knew that. And these internalization increase over time as expected, with very little internalization of nanoparticles in macrophages after 24 hours of perfusion. As said, if you look at the plot of internalization, the kinetics of internalization is very similar to what we reported before. What is really astonishing is the increase when we have in cancer cells that are cultured in dynamic, they do express possibly more CD44, and hence the internalization increase and pick up again. So that was very positive for us. So in essence, and I keep probably to be on time, thankfully. So the lesson that we learned from this very preclinical study stage was like we could effectively and efficiently design a nanocarrier that is able to protect RNAs, even against harmful agents like enzymes. They kept the stability over time up to 2 weeks. We could test for longer, but it's in the to-do list for the future. This nanosystem should guarantee a prolonged circulation in the bloodstream where injected. They would promote the release of sRNA in cell cytoplasm. We know that they do escape lysosomes and release effectively sRNA via imaging and by direct measurement of expression of mRNA level of a gene of target. And we also can learn that if we engineer better the in vitro model, we could learn more from the design of the system itself. So we proved that even in the presence of co-culture, in the presence of fluid flow, this nanosystem were effectively taken up and releasing their cargo in cancer cells rather than off target cells. So of course, I would like to thank all the people involved in these studies. It wouldn't have been possible if it was just myself, of course, mainly Professor Nicola Tirelli. Kay Williams was of help as well. A lot of thanks are going to Julio and Enrique. They're already well, graduated and working somewhere else. They left the university a few years ago, and Wia as well. And all the other industrial collaborators that had a really great input into this project. And I would like to conclude my presentation now to give you a bit of an insight of where we should go probably in the future, because the problem that we faced in this old research was due to batch variability. So we moved on, and now in my group, we are trying to look on the use of microfluidics as a new way to manufacture and control better the nanoparticles themselves. And at the moment, we work on two different projects. Sorry. One is looking at the fabrication of liposomes using a microfluidic chip, or to make polymeric nanoparticles as a microfluidic chip. These two researchers started a bit offset. So the one on liposome started a bit earlier than the test of this system to produce polymeric nanoparticles. And they both use the same principle. So same device, same chip, of course, different solution. What we look here in this chip and the variables of this manufacturing process are basically the material that you use. So if you select an FDA approved material, very well characterized and well known, you could really speed up all the paperwork to approve it for clinical use. So what we can control then is the viscosity of the solution, so the percentage and concentration of solution, of starting solution. We could have a control over temperature, and these would affect the mixing at the junction, as you can see in figure. The very first study that we did was comparing a traditional or conventional manufacturing process, so the well-known thin film extrusion method to make liposomes with a microfluidic system. And we made, for this very first study, a drug-free liposome, so made of DSPC, cholesterols, and PEG. And we looked at the effect of this flow rate, so how we would find tune the balance between the cross flow rate of the aqueous phase and the flow rate of the organic phase containing lipid. And as you can see, if we increase, so with TFR is total flow rate. So if we increase the total flow rate, we don't see a big variation on the size of liposomes themselves, but the big point is the dispersity. So the monodisperse curve of the size of these liposomes that we obtain becomes more narrow. So we have a very high control over the size of nanoparticles or liposomes at this stage. And on the other side, we looked at the cross flow, so the flow rate ratio. So if this ratio at a constant flow rate, the optimal that we identified earlier, would impact on the size of liposomes and the size distribution. And again, you could see from the plot that we could identify what was the optimal flow rate ratio to get a really high control over the size of about 100 nanometer. That was our target, and a very narrow distribution. So we don't see a big variation between all these nanoparticles. Same thing was done, well, we have done it for polymeric nanoparticles. We identified a ideal molecular weight of PLGA and a good surfactant, in this case is PVA. And we looked at, again, at the role of the flow, in this case, flow rate ratio on the first slide, and the total flow rate on the right, sorry about it. But so we noticed that if we increase the total flow rate, for the case of polymeric nanoparticles, we don't change a lot the size of produced nanoparticles. What plays a big role in size and size distribution in this case is the flow rate ratio between the organic and the water phase. Again, that's easy to explain. It's because in the manufacturing of PLGA nanoparticle, we have a nanoprecipitation, and this impact on how two immiscible fluids are mixing and how polymeric nanoparticles precipitate and then flow throughout the microchip. So I want to show you interesting results that we obtain on the liposomal bit. In particular, we screened, so in parallel to that study, we were screening and the possible use of coumarin derivatives as a co-administration drug with doxorubicin. And we identified in this research, out of a library of 21 coumarins derivatives, the one that had a higher effect on breast cancer cells. As you can see, IC50 values are not incredibly high compared to standard and first-line treatment for chemotherapies, but still they do have an effect on cancer cells. So the idea was to form a liposome that could load umbellipronin, the coumarin, in the lipid bilayer, because of this high hydrophobicity, and then doxorubicin in the core of liposome and use it as co-treatment. We compared again the two preparation methods, so preparing conventional thin film versus liposomes prepared with a microfluidic system. And as you can see, the size obtained was not very different. So in the thin film method, we could have an average size of about 150 nanometers, quite distributed. Whereas with a microfluidic system, we will have a slightly larger size, but very well distributed with a low PDI number. Drug encapsulation efficiency was comparable between the two systems. Yeah, I will finish. I'll wrap up in a second. And we tested that. The lesson that I want to show here is that when we started to include doxorubicin in the lipid bilayer, we improved dramatically the formation of liposomes. So the take-home message from this study is to really look at the rigidity of the membrane of liposomes and try to design and tailor well using quality by design method. What we obtained was a really nice study where we could compare the effects of doxorubicin released from liposomes with and without the encapsulation and coadjuvant of ubenlipranine. And the other study that I would like to show is we are at a very preliminary stage. We are preparing now the manuscript. So Shua is working hard on that. And what we want to look now is to develop a nanosystem polymeric system that is able to release the drug over a longer period of time. So not just within the very first 24 hours, but looking at something that would release the drug over a 2 weeks period of time, where we could inject locally as a depot system, such nanosystem, hence have a more compliance with patients. So to conclude, really, it's just, I think this is going to be probably the future. We will using more technological advanced devices, we could speed up and be more GMP compliant from the very first phase of designing nanoparticles. We could have a higher control of the manufacturing steps and possibly even reduce them. This would lead to obviously a reduced batch-to-batch variability, and this would also allow us to screen a larger number of formulations rather than one only. And again, trying to reduce the gap between preclinical and clinical stages. So I finished possibly on time. Many thanks again for your attention. And again, thanks to all my group that is producing and going back to the lab finally and being very highly engaged with science. And I hope you find this talk attention, and I'm here for your question. Thanks.
We have a couple of questions. Can we read it out?
Yeah. So what about the size of hyaluronic acid? Does it affect the overall size of nanoparticle? Is it possible to use any other sulfated protoglycan? Yes. So first thing, yes, the size of hyaluronic acid does have a big impact. I didn't show that study. It was a study done before myself joining this group. Yes, it was found an optimal size of about few hundred kilodalton. Smaller hyaluronic acid do not complex and are not stable on the surface of nanoparticles, and larger are just leading to really big complex that are not possible to be injected intravenously, so they were discarded. And yes, it is possible to use other sulfated protoglycans. Again, the question here is optimizing the ratio or between all the components of the formulation, and again, reach the target size and stability. I hope I've answered your question.
Yes.
The second one is, you would like to know how you evaluate the stability. So stability was tested in terms of size, surface charge, with using dynamic light scattering measurement. We also did some imaging. We haven't shown it here, with some AFM as well studies. we also looked at stability for the protection of the cargo, as I showed. Probably this question came at a very early stage. So we showed also stability and protection of the cargo itself. So stability of the efficacy of the nanoparticles. So not just the physical chemical part, but also the therapeutic part. So could be the significance of surfactant and co-surfactants in liposome preparation through microfluidic approach? Yes, it's important also to include those. The question here is how many variables you want to include into the system. So having already few compounds, as I said, we discovered just by doing experiments that umbelliprenin itself was playing a big role on the rigidity of the membrane, and that this would impact on the mixing of the miscible and miscible phase and lead to the fabrication of very narrow monodispersed liposome with a microfluidic system, and also would help with encapsulation efficiency of doxorubicin. That was just an experience we learned from the lab. So if you include another variable, of course, you need to screen many other things. Yeah, it depends on the capacity that you have on the study, probably. I see other questions. So there were, and then the question and answer in the chat. There are more questions. Well, thanks. Thank you for listening. Thank you for the informative talk. Please tell which material-based nanoparticle will be most effective in vivo. Good question. I think looking at, so we did this research on the nanomedicines that were approved over the past years. So probably over the past 40 years, I have in my head the liposome-based nanotechnologies are the most used and the one approved at the very early stage. I think it really depends on the application. There's very little use in the clinic of nanomedicines at the moment. So I guess there's still some open question and again, the need to prove that they're more efficacious than standard of care currently used in the clinic. But I guess, again, there are now many of the COVID-19 vaccines that use nanotechnologies as a vector, so probably that would boost a bit the approval of nanomedicines in the next future. I really don't know which one is the best. I would like to have to answer myself.
Thank you. Okay.
Thank you, Dr. Annalisa. It was a pleasure hearing you. We also thank you for your time and effort. Have a great day, ma'am.
Thank you so much.
Thanks, and thanks again for inviting me. It was a pleasure. I hope there will be a chance in the future to visit and see each other in person at the conference again. Thanks again and have a good day.
And now we come to the end of today's lectures. Hope you all had very informative and engaging sessions. We'll resume with the remaining talks tomorrow 9:30 a.m. IST and continue as per our schedule. Please make sure you join on time. Thank you for all the participants. Have a good day.
Sudha?
Yes.
Okay, we can meet tomorrow.
Okay.
Okay?
Okay.
Tomorrow 9:15 we can start by 9:30, but the online we can start by 9:15.
Sure.
Okay?
Sure.
Madam.
Yeah, sure.
Okay, okay. Thank you so much. Okay, see you.